So today's discussions will be on basically on how to predict the different type of promoter sequence with respect to any gene, and how to find out along with the promoter different regulatory element sequence. So as all of you know that when the protein is expressing, we require the mRNA, and the mRNA is going to produce from the DNA itself, process called transcription. And during this transcription, enzymes of course required RNA polymerase, and that is having variation for eukaryotic and prokaryotic system. Now, how RNA polymerase is doing function and what are the basic sequence definitely to be present when the transcriptional process is going on, and how by doing the in silico method or in silico process we can able to find out those particular sequence. So that I am going to discuss today. So for expressing the gene, how the promoter is going to be find out, and along with the promoter or to controlling the promoter, what are the other sequences are mandatory and which are having direct influence on the promoter, and therefore the polymerase will continue the functions. So this will be our discussion today, and I'll be also showing that how to use the in silico process to finding the promoter as well as regulatory element with respect to the any gene sequence. So let's start promoter and the regulatory element prediction. So promoter, as of we know, so this is a particular sequence which is to be present in the DNA level, and by looking that particular sequence, RNA polymerase and all other accessory factors whatever is required, that is going to bind, and finally which is called the starting positions. Conventionally it is called the +1 positions, where exactly the transcription is going to start. And here whatever the discussions I'm going to talk, so that is all depending on the mRNA, how mRNA is going to produce, because in the biological system, as of you know, that mRNA only can able to produce the protein, not the tRNA or the rRNA. So they are having different function, and how they are synthesizing in the biological system, that process also called transcription. But here our main discussions will be on the mRNA synthesis and how to predict the mRNA promoter for synthesizing the mRNA, how to predict the promoter, and how to finding out the regulatory element, that is the TAPBs, different transcription factor binding site, how we can able to analyze. So this, without the help of the promoter, transcription process is not possible to continue, and that is truth for both eukaryotic as well as the prokaryotic system. So this transcriptional factor, these are group of protein along with this particular promoter, along with this RNA polymerase, this is also important. So all these things together helping this RNA polymerase to do its transcriptional function. That means it helped to RNA polymerase to continue the transcriptional process. So you have to understand that for prokaryotic system here, and you need to know for prokaryotic system what type of promoters are there, which is always it will be written in the biochemistry and molecular biology genetics book. This is the promoter sequence, but it is not always the truth, because we can see a lot of variations are there in the with respect to the different gene in that promoter level of that particular sequence. So all the things, how basically by applying the in silico method and what kind of algorithms are there behind that, basically I am going to discuss on that only. So there are having traditional molecular biological concept to finding the promoter sequence, and the process is called DNA footprinting experiment. So here where the RNA polymerase is binding, so the restriction enzyme if you are using, so as the restriction enzymes are not able to present because of RNA polymerase it is hiding, so that particular part is not going to cleave off, and that sequence is going to give the quite lengthier conditions comparative to the control sequence. And by looking the gel pattern, we can understand this is the particular area where the promoter was able to cover, and that's why the RNA polymer is not able to cleave off. So this experiment is the common molecular biological experiment called the DNA footprinting process. So we are taking the help of DNA footprinting that experiment. We kept the idea and the sequence, whatever according to the experiment has come out, we are keeping in our mind. And based on that, we have designed the algorithm, we have constructed the algorithm, and perfectly in silico method, it is going to detect such kind of sequence, which is considered as the promoter. And a lot of softwares are available for that, and how to handle the software that we are going to understand today. So if you are determining by in silico method these promoter sequence or different type of regulatory elements, so a lot of problems actually we are facing. So what are these problem? Problem number one, that is the promoter and the regulatory elements are not clearly clearly it has defined, and therefore we can tell this is the highly diverse sequence. So when the lot of variations are there in the sequence, then it will be very much challenging process, or it will be very a kind of challenge to all kind of algorithm designer. So it will be problem for them to prepare a single type of process or single type of step. So by if you are using the step and if you are following the single algorithm, we can able to predict it. So it is a kind of challenge. So how we have overcome the challenge, that I'm going to talk today. So this was the first problem basically when starting algorithm people have faced. And next important problem whatever is there, that is this promoter and regulatory element cannot be translated into the protein. So from mRNA, DNA to mRNA, when it is producing this promoter sequence or that regulatory sequence, it is not coming to the mRNA. And as it is not coming to the mRNA, definitely it is not going to come to the protein level. So that means if it is there in the protein, so we can analyze the protein, and from there we can easily able to understand that which particular sequence is actually promoter and the regulatory sequence. So as it is not translating into the protein, so that is also one kind of difficulties to prepare kind of algorithm. And third important problem that is we are facing when we are preparing it, that is the promoter and the regulatory side to be predicted are normally very short. The sequence is very short, hardly 6 to 8, or some cases it can found that 10 also. But yeah, this small particular segment of the sequence, and that sequence is having a lot of diversity, that means variation, and therefore we are not able to initially apply a single kind of algorithmic approach to identify the promoter as well as the regulatory sequence. So these three was the major challenge for the computer science people, and they have taken this challenge and they have prepared kind of algorithm, and now we are using the algorithm to finding the specific gene sequence as a promoter and some regulatory sequence too. So here by looking this kind of problem, and when they have tried to solve it, they have designed a 3 different type of algorithmic approach. One is called ab initiation or initio based prediction approach. Second one, prediction based on evolutionary information, and third one is the experimental data approach. So these are the 3 different way are there. So different software are open source, they are using the different method. Either it can use the ab initiation based method, otherwise it can go for evolutionary confirmation or information approach method, or some server they are using the experimental data approach method. But experimental data approach is not so much familiar, because why it is not so much familiar, I'll be telling. So here is having maximum drawback. So by looking those drawback, so we are not preferring for any kind of research to use such kind of approach. So promoter and the regulatory element in the prokaryotes. So if you are talking about the prokaryotic promoting elements and the different regulatory elements, so for that you need to understand better way how actually these transcriptions are happening, and how basically these hello.
Yes, sir, we can hear you.
it is raining outside, some problem I think happening. Can you hear me? Hello?
Yes, sir, we can hear you.
Oh, okay, fine. So here first you need to understand that what is the molecular basis to continue this transcriptional activity in the prokaryotic system as well as in the eukaryotic system. So for prokaryotic system, if you look, there is having a single RNA polymerase enzyme and which can continue the all sort of transcriptional process, all sort of gene to be transcribed with the help of this single polymerase enzyme. And RNA polymerase for prokaryotic system, it is the multi-subunit enzyme. So many subunits are there, and the subunit will be joined together to make the functional this particular promoter and functional this particular polymerase enzyme, and then it is will able to do the its correct function. So this RNA polymerase, if you look, there is one of the component of it is the sigma. So we can tell it is the sigma subunit. So majorly for RNA polymerase, maximum cases for prokaryotic system we can found is as a sigma 70. So 70 stands for, this is the molecular weight of the sigma protein. So this is the major transcriptional factor we can tell, or we can tell this is the major component for the RNA polymerase rather than transcriptional factor, major component of RNA polymerase. And this component only can able to identify the sequence of the promoter, and it first go and bind there, then other subunit one after another it will assemble, and finally the infrastructure of the RNA polymerase will produce, and then RNA polymerase can able to do the transcriptional activity. So here this sigma factor can find out in the all kind of prokaryotic system. If you look, basically they can find out that particular sequence, that sequence which is considered as the promoter, and that is finding basically 35 base pair region and 10 base pair region. This is away from the starting site. And if there is having convention that if where exactly the transcription is starting, that is called the +1 point, and its right hand side, the gene which is going to transcribe, that is considered as the plus site, and the left hand side, that is where the promoter and all kind of regulatory sequences are present, that is considered as the minus site. So if we look at from the exactly from the starting positions, we can found -35 region, this particular one sequence is present, and -10 region, that another particular sequence is present, and both the sequence in this particular region are very important because sigma factor can bind the can able to find out this sequence, and until unless it is going to find out this sequence, gene is not going to transcribe. So this -35 region, that particular sequence whatever is present, so maximum prokaryotic system it is having this sequence TTGACA. So we can tell sometimes this is the -35 box. At the same time -10 the region, this is having TATAAT. This is the sequence, and this sequence is called the -10 box. So this is the in molecular biological term concept, we can tell this is the particular site or the motif where these sequences are very much present for all kind of prokaryotic system. And this example of sequence is basically found in the E. coli. So if you look at the other group of bacteria, might be the sequence 1 or 2 alphabet to be changed. But as the common cases, we found these are the conserved sequence, or these are the common sequence. So we are giving the name -35 box and the -10 box. Now, this sequence, this as I mentioned, sigma is going to find out, and ultimately once it is binding, all other factor will be coming to close and making the functional RNA polymerase, and that is going to continue the transcriptional process. Now, apart from these RNA polymerase and all these subunit of RNA polymerase and these promoter site, there are required other various kind of proteins. And without the help of this protein, RNA polymerase and all these particular promoter site will not able to continue the transcriptional process alone. So those protein which are helping in these cases, so they are called the transcriptional factor protein. And this transcriptional factor protein we can found both for eukaryotic and the prokaryotic system. And this transcriptional factor protein help to sigma factor to find out this sequence, and therefore the sigma factor is binding, and they're also responsible to assembling all other component of the RNA polymerase, and therefore functional RNA polymerase is producing. So we need to understand that what is the role of this transcriptional factor and how these transcriptional factors are important to controlling actual this gene expression in the prokaryotic system. This transcriptional factor, whatever is there, so they also can bind to the specific upstream region of from the promoter, and once they are binding there, they are controlling the promoter sequence activity, they are controlling the RNA polymerase activity too. So this regulatory element, so we can tell this transcriptional factor where they are binding. So that particular part is called the regulatory element site, and this site also is having a lot of influence to control the transcriptional activity as well as the promoter activity. So the reason that the regulatory proteins binding from the away from this particular promoter site, and we know that the DNA can go for different type of folding process. So this, when the DNA is melting and going for folding process, so that time this regulatory element, all kind of transcriptional factor, RNA polymerase, all are coming closer and making a contact with each other, and therefore the functional transcript complex is going to ready, and that's why the RNA polymerase is pushing or moving towards the your starting site of the transcription, and finally the gene is going to transcribe. So this is the -10 box, this is the -35 box. So this is the RNA polymerase is binding, and apart from that, the TF is indicating this is the transcriptional factor which are present towards another upstream region from the this promoter also. So once these proteins are binding, DNA is melting and folding, and because of that, these are making a contact with the polymerase, and finally from the transcriptional starting site, the gene is going to transcribe. So this is the normal molecular biological aspect we can see, which is going to continue in the prokaryotic system. So promoter and regulatory element in the eukaryotic system, if we compare with the prokaryotic system, we can found some similarity. Of course it is there, but as the eukaryote is having more complex genome, and eukaryote is having different type of gene in the genome, so the process will be bit complicated, of course, than the prokaryotes. So you need to understand that again, how the eukaryotic gene is going to express, and what kind of mechanisms basically are continuing in the biological system to expressing any kind of eukaryotic gene. So promoter and regulatory elements in the eukaryotic system. So here, apart from the promoters, we can found in the eukaryotic system actually 3 different type of major polymerase enzyme. Of course, RNA polymerase I am talking. So polymerase 1, RNA polymerase 2, and RNA polymerase 3. Among these 3 polymerases, RNA polymerase 1 and 3 are responsible. 1 is responsible for encoding ribosomal RNA, and polymerase 3 is responsible for tRNA. Whereas polymerase 2, it is the vital RNA and RNA polymerase, and these enzymes are helping to making all sort of mRNA in the eukaryotic system. As the beginning I mentioned, our discussions will be on the mRNA, how it is going to synthesized, and there how these RNA polymerases are responsible, and of course the promoter sequence, how it is there or how it is controlling, along with the different type of regulatory sequence, how they are influencing to the promoter, and therefore RNA polymerase is controlling the activity or doing the activity. So that is our major concern. So I'm not touching this polymerase 1 and polymerase 3. So right now there is no particular algorithm or the open source or the purchasable software which able to find out the promoter sequence for the RNA polymerase 1 as well as the RNA polymerase 3. At the same time, they are not having any kind of software which can able to find out the regulatory sequence related to the RNA polymerase 1 and RNA polymerase 3. So that's why our main concern will be there to how these mRNA are going to synthesize in the eukaryotic system, as well as how it is going to synthesize in the prokaryotic system. So this is the first complexity we can see in the eukaryotic system, that we are having 3 different type of RNA polymerase enzyme, and all the RNA polymerases are having different kind of function. And of course, each eukaryotic gene have in its own different type of promoter. Fine. Whereas prokaryotic gene, that promoter concept, whatever I mentioned, -35, -10, and they are almost what are the sequences we are found, and those sequences almost means will be the conserved. 1 or 2 alphabet can be changed, and hardly 6 to 8 sequence I mentioned to be present in the promoter region for the prokaryotic system, and some cases might be it will be 10 to 12. That is the only difference we can found, but maximum cases we found that is -35 having that sequence, conserved sequence, and -10 is having that is the proper conserved sequence. But if we come to the eukaryotic system, every gene, eukaryotic system is having n number of gene we are having, and n number of genes having n number of related promoter sequence, and the promoter is going to control the gene function. So we need to understand with respect to the all kind of gene. Okay, and the transcriptional machinery also required the many kind of transcriptional factor. So prokaryotic system hardly require 1 or 2 different protein as a transcriptional factor containing the all activity, but for eukaryotic system here n number of proteins will be there, and all the proteins one after another sequentially will work, and finally they will be controlling the promoter's activity, and that way they are going to control the RNA polymerase II's function. So RNA polymerase II here also I need to understand that it cannot able to directly bind to the different promoter part which is present in the DNA level. Fine. So for binding also it required a lot of transcriptional factor. So for binding also required factor, to continue the transcription also RNA polymerase II eukaryotic system required lot of specific factor, and these factors sequentially will be working, and that way RNA polymerase will be functional. And basically if we look at the eukaryotic core promoter sequence, so there also we can get so-called TATA box, okay, Tata kind of box or Tata box, and it is also located like 35 base pair away from the upstream region of the transcription, and that region also maximum time we can see this Tata, though we are telling this name, but they are actually having before and after some another few nucleotides, and all these nucleotides are important, and that's why we can tell this is the motif part of the DNA where the RNA polymerase can bind with the help of different transcriptional factor. And as I am telling, this is the motif part because we can see a lot of variations. So TATA along with some gene it will be present A, and some gene it will be present T, and some cases it will be whether A and T, after that definitely A will be there, and again some difference we can get with respect to the some gene, again A or T. So as this variation you can see with respect to the different gene, so we are telling this is the motif part of the transcriptional site. And though the Tata, this name we are telling because the TATA, this could be the common for all kind of promoter sequence, but rest of the alphabet whatever is there, so that is going to change. That's why it is the motif. And here as I mentioned, lot of transcriptional factor required. So this transcriptional factor along with the RNA polymerase, there will be having a linked, and this linked will help to RNA polymerase to transcribe this particular gene sequence. So here that a lot of regulatory sequence also to be present. So those regulatory sequence like suppressor sequence, like activator sequence, etc. We give this kind of name. So because activator sequence, when activator kind of factor is binding, so it is activating the RNA polymerase to continue the transcription. And suppressor system, when the suppressor protein is expressing, it is controlling the activator site, and that's why the RNA polymerase transcriptional process somehow it will be halted. So these are the sequence are considered as the transcriptional factor binding site as well as the regulatory sequence. And for eukaryote, this regulatory sequence for all kind of gene is bit different. But how it is working for all kind of gene, the pattern will be the same, but sequence of this particular factor, how it is binding, that process is same. What is the only difference? Difference is which gene is going to express. With all the genes, this activator part may not be there. With all the genes, the suppressor part may not be there. So the regulatory site wise, the length of the regulatory sequence and the function of the regulatory sequence and the molecule which is going to bind to the regulatory sequence, that would be the different. And also we can get the difference in the promoter sequence level. So Tata is the common, then next all the alphabet, so with respect to the gene, it will be changed. So this is the way or this is the common picture you can get after the molecular biological experiment we came to know. So these are the common picture. So how this RNA polymerase 2 along with all transcriptional factors are together doing the function in the eukaryotic system. And here also we need to remember that not all the eukaryotic system, whatever the genes are there, they are having the data box. So at beginning I mentioned, so this sequence is not the conserved, fine. And for prokaryote, almost it will be like a 99% it will be conserved, but for eukaryotic system, it will having lot of difference. Fine. So here with respect to the data, this sequence whether present in the common gene or not, I just mentioned. But if we analyze the entire gene part, whatever found in the eukaryotic system, there we found this not all the genes are having this kind of data box sequence. So especially the housekeeping genes, so definitely they do not have this data box sequence. So these housekeeping genes, these are vital genes which are expressing continuously in a constitutive fashions and maintaining all kind of important biological activity, biochemical activity, physiological activity in the eukaryotic system. So we are having n number of genes for controlling this, having this housekeeping function. So all the genes are also again having this different kind of promoter element or the promoter sequence. So this is also one kind of challenging events to designing a particular algorithm to understand the function of this particular promoter. And if we some other gene also you can see, especially this housekeeping gene, if we if we analyze them, so we can found along with this particular gene there is having another kind of sequence called the unique initiator sequence, that is the INR sequence. And this INR sequence basically having this pyrimidine rich one part, and that part is the again the consensus part. So pyrimidine this part we can get either having C or T, or then again is having C or T, then C and T definitely we found, and again there will be having variation either C or T, then again C and T. So these variations again we are getting, though we are telling this is the consensus. So consensus with respect to telling these are the only variations we are getting, but this is not the actual or truly in the sense of consensus. Okay, so these are the variations we can get in case of the housekeeping gene in the eukaryotic system. And transcriptional factor also where it is binding, that positions wise also we can find the difference in the eukaryotic system. So on an average there will be having 500 base pair basically sequence, and that 500 base pair sequence to be present away from the promoter part, that is the minus part with respect to the gene, it will be present, and this transcriptional factor can able to bind there and ultimately control the promoter's activity. And eukaryotic system, almost this 500 base pair, it is almost equivalent to your with respect to the molecular weight, it will be the 5 kb or the 6 kb, maximum 10 kb region. So within the 10 kb region, we found almost all kind of promoter sequence and controlling sequences are present to the upstream part of the gene. And so 1000 of base pair away from the starting site, if we consider 500 base pair, of course it will be 1000 to 2000 base pair away, but within that particular region only, with respect to the housekeeping gene and all other kind of gene, we can found the promoter regulatory sequences are present. And occasionally regulatory element are also we can found with respect to the RNA polymerase 1 and RNA polymerase 3 promoter, the transcriptional this regulatory sequences are present downstream part of the gene. So at the beginning also I mentioned that there is a no proper your algorithm so far is available. By using that we can able to predict the downstream part what are the regulatory elements present in the particular gene sequence when it is going for transcribing. Fine. So here this word is indicated with respect to the type 1 and the type 3. So for them some regulatory sequence we can found also to the downstream part. So and here our one advantage out of by looking all these particular mechanism, one advantage or one particular conclusions we have drawn, and the conclusion is like this, that there is having a clustering region. So why I'm telling this clustering this word, because these regulatory factors and the promoter wherever it is binding, the RNA polymer is itself a protein, and all the regulatory factor also a kind of protein. So when a maximum proteins are binding with respect to the expression of gene, so that particular cluster part we are giving emphasis. Okay, so by looking that cluster part, we have designed the algorithm. So this clustering part we have able to understand by looking this wet lab experiment. In the wet lab, already these data, some people have proved this is the site, this is that sequence, some proteins are binding, and ultimately they are controlling the RNA polymerase function. So this clustering concept we are applying to finding the promoter as well as the regulatory sequence, both in case of the prokaryote as well as the eukaryotic system. Okay, so this clustering approach is very important. So you are able to understand that normal molecular mechanism. So how this regulatory sequence and where the regulatory sequence will be, what kind of sequences must be present in the both prokaryote and the eukaryotic system with the respect to the gene, how the sequences again varying for the eukaryotic system, etc. Now I'll be talking about the what kind of algorithms we are using for predicting this kind of sequences with respect to the gene, right? So promoter and the regulatory element prediction algorithm. So here we are having three different type of approach or algorithm. One is called AB initiation based, second one is the similarity based, and third one is the expression profile based. Whereas AB initiation based, it is basically de novo, it is tied to scanning the individual sequence. So whatever the sequences we are providing, and the sequence, every alphabet it is going to analyze, and with respect to analyzing that particular sequence, it is going to predict this could be of the regulatory site, and this could be the promoter site present in the upstream part of the sequence. And second approach is the similarity based approach. In the similarity based approach, it is based on the alignment of the homologous sequence. I repeat, it is based on the alignment of the homologous sequence. That means in the database some information is there, my sequence will be telling with that, and I'll in the database with respect to the sequence, whatever the information already available, this is with respect to this sequence, this is the promoter site, this is the regulatory site. So if you have found the provided sequence is perfectly matching with that database, that particular sequence, then we are predicting my sequences also having such kind of promoter element and the regulatory element. So that is called by this way, basically once we are telling the sequence, we are trying to find out the alphabet to alphabet what kind of similarities are there. So this process also called phylogenetic footprinting method. So DNA footprinting, that is wet lab method, which is predicted that exactly where the RNA polymerase is binding. Where in the eukaryotic system, first this experiment have done, and there, as the name has given, DNA footprinting to identifying the RNA polymerase binding site. Here it is called the phylogenetic footprinting, because as the similarity we found in the phylogenetical level with all the sequence, how we are getting the similarity, and based on those sequence related informations, whatever is available, we are adopting. So same concept we are putting over here. So this is called similarity based approach. Also, the name is phylogenetic footprinting approach. And third one method is the expression profile based approach. Here, expression profile based approach, again, this is also depending on the wet lab experiment, but this experiment is completely depending on microarray data analysis process. That means that microarray, whatever the informations already we are having. So microarray basically is talking about the RNA expression pattern in the particular cell. So we are going to compare with the all RNA expression pattern of the cell along with the my mRNA sequence, and based on that we are telling. So as those sequences are having such a so and so promoter, so my sequence also might have similar kind of promoter. So these are the approach in algorithm designing we are following to finding the different type of promoter and the regulatory element with respect to the eukaryotic system and also for the prokaryotic system. Fine. So all this algorithm, again, I am telling it is depending on only the mRNA polymerase 2 for eukaryotic system and all RNA polymerase, only single RNA polymerase found in the prokaryotic system for both of them. Okay, so ab initiation based method, similarity based method, and the expression based method. So first I'll talk about the ab initiation based method. In the ab initiation based method or algorithm, so there is having particular consumption assumptions. What is the assumptions? This particular method is completely depending on the signal based. Okay, this ab initiation program is completely depending on the signal based. So signal based, what kind of signal? So signal is indicating the characteristic promoter sequence, that is a Tata box, whether present or not, because the sequence alphabet, whatever I mentioned just before. So that kind of sequence, whether in the provided nucleotide it is present or not, that is first approach, first point. And second point, it is also rely on the content information such as the hexamer frequencies. So what are these hexamer frequencies? Hexamer frequencies is the starting codon, and the basically there are having 3 starting codon. And if we talk about the DNA, so both the stand 3 plus 3, total 6 reading frame will be there. So that's why all the 6 putting together called the hexamer frequencies. So how better way these hexamers are appearing? That means the starting codons are appearing in the gene level or the RNA level, so that these 2 important parameters in the ab initiation based method basically try to find out, and that's why it is called the signal based approach also. And this advantage of this method is that the sequence can be applied in such a way that which is having obtained the experimental information. That means experimental information already we are having, wet lab informations we are having, and that information basically we are keeping in our mind and try to put into the in silico method too, and we are going to find out that particular alphabet, whether it is matching and it is present or not. So this type of method, it is now they're also having some kind of difficulties. The difficulties, it is very difficult to discovering the new motif or the new sequence if it is present in that respective provided gene sequence. So we are expecting, or we are setting the algorithm like with the Tata box and the starting reading frame. But if the Tata box, some alphabets are going to change. That's what I mentioned before. So 8 to 10, the variations we can found itself in the prokaryotic system. So that variations, this may not able to predict perfect way. And that way, sometimes it can miss the some kind of motifs where actual these promoter elements are present. Okay, so to overcoming this problem, or what basic conventional approach you have taken. So the conventional approach, there is having 2 different concepts. Okay, so as the ab initiation method, the initially whatever the assumptions we have taken, based on the assumptions we found some kind of drawback. To overcoming the drawback, these further assumptions we have, or further approach we have taken, and now by looking this approach, 99% we are able to predict the perfect promoter and the regulatory sequence. So what are this new conventional approach? So new conventional approach is completely detecting a promoter or the regulatory site is throughout the matching of a consensus sequence pattern. So it is not only consisting on the Tata box, it is tried to find out wherever in the sequence with whom we are comparing. So wherever the between these 2 sequences are having the consensus sequence. Okay, so enter consensus sequence pattern and the total length of the consensus sequence they are going to analyze here. And they found if between this consensus sequence, if anywhere this Tata, these compositions are present, so the software is predicting might be this is also promoter site with respect to this particular gene. Number 1. Number 2, also some cases we are applying the matching position specific scoring matrix. So when we are with respect to a particular score, we are telling now this particular consensus sequence how much score is giving. So based on that, we are telling also this is the perfect that particular promoter site, or the perfect consensus sequence matching is there, and that's why we are the software, whatever the result it is predicting or providing to us, we can believe on that. Okay, so the PSSM, that is the position specific scoring matrix process we have introduced here. Okay, so I'll be talking on that particular PSSM also, how it is working. So this consensus sequence or the matrices are relatively very short, and as I mentioned, this promoter sequences having hardly 6 to 8 that particular alphabet, okay, nucleotides. So it is once again, it is very difficult to find out among the thousand or billions of base pair where exactly these 6 or 10 bases are present. So to finding or to understanding this particular concept, we have introduced this PSSM pattern. That is the position specific scoring matrix, this particular pattern. And here, this pattern, how you are selecting, that is called the weight concept approach. So when we are talking about the weight concept approach, definitely here Markov models and the hidden Markov models both to be applied along with the artificial neural network. So in the gene, how to predict, there I've explained already what is the Markov model and hidden Markov model, how it is working based on the conditional probabilitical approach. And also I mentioned there what is the artificial neural network and what is the meaning of the giving input, then hide layer and coming to the output. So same approach here also to be applied, and but keeping this thing in our mind that we need to ultimately give the score with the respect to the PSSM, that is the matching, that is the position specific scoring matrix process. And this neural network or hidden Markov model, whatever you are using, this based on this, whatever the algorithm we have designed. So this algorithm, some of them are telling, so some of the algorithms selectively exclude the coding region. Our concern is only with the finding the promoter region, finding to the non-coding region as such. So here they are excluding the coding region. So the region which is actually WRF is present, which is going to protein. So that particular part they are excluding. So that way they are shutting down the particular region which are not expressing, and analysis will be much better and easier with respect to that particular part. So these are some of the algorithm here, whatever the neural network or hidden Markov model we are using, that's the concept we are keeping in our mind and continuing this process. And second approach also, it is there for some cases we have focus on the upstream region. That's before I mentioned within 500 kb maximum we can found this particular upstream regulatory sequence are present. So that particular region only we have taken our consideration. Okay, so this way these two process help us to better way understand with the ab initiation based method to where exactly and how much the regulatory sequences are present and where exactly that promoter sequence and the starting site is present with respect to the gene. And is this way, if you are considering this particular 500 kilobase pair, that particular region from the upstream, so the approach is now both the promoter as well as the regulatory sequence together or the coupled way we can able to find out. So I'm going to give in the practical session whatever I'll be showing here, so you can understand how these two are working together. So you'll get the better way, the idea. And the ab initiation based method, I repeat, ab initiation based method till now is the best method to identify this regulatory and the promoter sequence from the particular gene. And next method, that this ab initiation based method, how we can able to apply for the prokaryotic system, how we can able to apply for the eukaryotic system. So first I'll be talking on the prokaryotic system, then I'll be coming to the eukaryotic system. For prokaryotic system, when we are applying this ab initiation based method, here completely the prediction will be depending on the operon structure to find out. So because of the prokaryotic system, we can get the operon and we can see the single gene itself or single site or single polymerase. That's what it is going to encode all kind of proteins like that. This concept we are having. And even with the operon, if we come to the prokaryotic system, so different type of gene also simultaneously is going to express from one particular starting point, right? So that's why in the prokaryotic system, when we are applied this ab initiation based method, our idea is to find out the promoter, the operon elements, how many operons are there, and exactly with respect to the operon, we are predicting the promoter as well as the regulatory part. So this operon prediction is the key in prokaryotic promoter prediction concept. So once an operon structure is known, it will be considered that only the first gene is predicted for the presence of the promoter and the regulatory element, because once the process is going to start, until unless entire genome or entire part or it is going to transcribe, So it is not going to stop. That's what we have seen in the maximum cases for the prokaryotic system. So that's why if you are taking the consideration for the first operon, so the promoter and the regulatory elements, whatever is going to find out, that is only related to the first operon, and we are not giving importance whatever the next operon it is there, and simultaneously whatever the gene is going to express on that. So first operon, we are predicting with respect to the first operon that promoter elements and the regulatory elements to be find out. Whereas other genes in the operon do not possess such DNA element, so that when the other genes are expressing, when the link part is there with the promoter operon, so there as the operon is already start working, that genes already start transcribing. So rest of the genes whatever simultaneously going to transcribe, they are not required such kind of elemental sequence. So that's why to finding the first operon and with respect to that particular promoter region and the regulatory region, to finding is the actual prime important for the prokaryotic system by using the ab initiation based approach. And for prokaryotic system, as per the Wang et al 2004, okay, Chinese people, so as per Wang et al 2004, till whatever the predictional process he has mentioned, and he published the paper in the Nature journal. So whatever the process he has mentioned till now, we are following for all kind of ab initiation based algorithm approaches. Okay, so what he has mentioned, the method whatever he has explained, the method is completely relies on two kind of information. One is the gene orientation, and second one is the intergenic distance of a pair of the gene of interest and conserved linkage of the gene based on the comparative genomic analysis. So he has given the approach like gene orientation to be taken care when we are finding that promoter elements or regulatory element. At the same time, the distance between the starting site and to whatever the other elements are present, so that particular site of the gene is also an important parameter here. And after finding these two, there are scoring pattern to be applied. So here SP that's position specific scoring metric pattern will be applied, and finally that way the entire promoter element to be find out for the prokaryotic system. And here when he mentioned this particular SPSSM position specific scoring matrix to be used, there also he has given us some kind of assumptions. So that assumption is like this. So according to this Wang et al method, so for two adjacent genes that are transcribed in the same orientation and do not have the independent transcription termination signal in between. What is the meaning of it? That means once the first operon is started transcribing that particular gene, so it is going to use the same RNA polymerase, and successively next whatever the genes are there, it is going to transcribe. But in between whatever the genes is going to transcribe, so that those genes should not have, or basically not having, any kind of stopping codon. That's what the first assumptions, and based on this assumption he has made this particular scoring pattern. So by keeping this particular assumptions, what scoring pattern he has mentioned, if the intergenic distance is greater than 300 base pair, regardless to the gene of linkage pattern, that means when the linkages are happening to the gene, which particular positions the linkage is happening, that means linkage concept, that means the physical mapping of the gene also he has basically taken care. Okay, and according to that, if the 300 base pair, this particular distance, if it is there between the intergenic distance, or if the distance is greater than 100 base pair they found, so that time he has mentioned the score should be 0. Okay, so this will be the first score of the giving that based on this particular distance pattern, the score should be 0. Next, if the intergenic distance is greater than 60 base pair and the linkage is shared between the less than 5 genome, then the score should be 1. So linkages can be between the 5 genome. That means when we are going to predict, definitely it is going to compare, doing the multiple sequence alignment with all kind of genome data, whatever is available, and between them the comparison is going on. And that way they found if the distance is varying like 60 base pair and less than 5 genome, we can find this kind of linkage distance is differing. So that time the score should be given 1. Like that, if the distance between the two gene like 32 between 60 base pair and the linkage is shared in less than 5 genome, or if the distance is between 50 to 300 base pair and the linkage is shared between the 5 to 10 genome, in both the cases the score should be 2. Like that, if you just look at here, the base pair sequence sometimes is vary, or the linkage pattern, the distance between the linked gene, that also sometimes could be vary. So here linked gene, why I'm talking, because in the genetic recombination and some kind of expression, we can get eukaryote as well as the prokaryotic system, and exactly gene mapping, that concept, it is not fully gene mapping concept. It is tried to find out that linked gene in the sense, in the meaning of linked gene is when the multiple sequence alignment is going on. If they found the alignment somehow left hand side and right hand side for some gene, it is moving. That means if they try to find out A should be present in all sequence in the same position, and if they found A is somehow is moving either left hand side or the right hand side for few sequences, so they are going to count that particular deviation. How much they have deviated position wise in from left hand side and the right hand side. That is the concept of the linkage. Okay, this is not actual that chromosomal linkage what is happening in the eukaryotic system. It is not like that particular concept. And if the intergenic distance is less than 30 base pair and that is having some kind of conserved linkage pattern, that means it is almost common for all nucleotide, the same position, same AAA, same position TTT, it is present. So, or in some cases, if they found that at least the 10 genome is having similar kind of matching. So all the cases, the score should be 3. And the last scoring pattern what they have mentioned, a minimum score is 2 if we consider the cutoff or the threshold for assigning the 2 gene in one operon. So both the gene, if it is present in the single operon, in such cases they have considered the score should be 2. If we look at this particular picture, it will be clear much more. So the scoring pattern we are getting less than 2. 2 is the threshold score. Okay, so threshold score need to go whoever is crossing, so that to be considered, and whoever not able to cross the threshold score, that is not going to consider. The score should be 0 when the D is greater than 300 base pair or D is greater than 100 base pair. That, and of both the cases the score should be 0, or the score should be 1, the 60 base pair, and the genome should be less than 5 is matching. So in that case, this is the negative part, and the line is indicating this is the threshold, and one is crossing the threshold, we can get the positive value, and by looking the positive value, we can consider this is the perfect consideration or the perfect way we have find out. So what is exactly the what is exactly the promoter part, and what is exactly the regulatory element, and how long these regulatory elements are present with respect to the prokaryotic gene. So this is the ab initiation based approach, and many open source, some of the purchasable sources are there. So maximum of them, it is not going to work out nowadays. I do not know why, but 1 or 2 definitely it is going to work out. PB-Prom I'll be giving as an example how it is working and finding the promoter sequence of the prokaryotic system with respect to the operon. And same concept prediction of eukaryotic system if we come. So here also consensus sequence based approach, basically it is following. So matching the consensus pattern of the known promoter and the regulatory element, this is going to tally, and based on that they are going to provide the score. And here this CPG island, that means some promoter site for the prokaryotic system, there is having a lot of variation, especially the housekeeping gene, and such cases the sequence of the promoter, and along with that some left hand side and the right hand side few nucleotide sequences will be there, and those nucleotide sequence along with this particular original sequence, that entire stretch is going to consider here, and that way the accuracy will be coming for predicting the eukaryotic sense promoter. And the eukaryotic transcription initiation required the cooperation of the large number of the transcriptional factor. So cooperativity means this is also indicating the clustering approach, where a lot of proteins are going to bind in one particular place when gene is going to express. So that is going to find out where maximum protein it is going to bind with the specific position of the DNA. So wherever the maximum protein is binding within this 500 base pair or 1000 base pair of the gene, So we are considering most probably that particular region is the regulatory region for transcribing any particular segment of the genome towards the your downstream part. So that way, basically, this approach is going on. And here also having GPG Prod, etc. These are all different open source, and all those are working basically hidden Markov model and your this artificial neural network process. Second different type of algorithm, it is the phylogenetic footprinting based method. So phylogenetic footprinting based method here, again, it is going to compare with the actually homology based method. This is so whatever the sequence I have providing, it will going to compare with the database all kind of sequence, and with those sequences, whatever the promoter and regulatory informations are provided, so that sequence information directly we are putting or we are predicting with respect to our sequences. So that's why it is called the phylogenetic footprinting based method. And here we are considering the evolutionary distance. We need to few things we need to remember here. So how we can select the database sequence with whom I supposed to compare. Okay, if you are taking the human and chimpanzee, and if you are comparing, and with respect to that, whatever the result output is coming, according to this particular method, it is going to give you that more false positive result. Why I am telling it is going to give the more false positive result? Because as we already know, the chimpanzee and the humans, the genome, if we compare, it will be almost 99.99% matching. So in such cases, we are going to get the similar result only. We are not going to get any different type of result. That's why more false positive, false positive result we are going to get. That means the analysis is not proper. So you need to consider when we are comparing between the two genes to finding out the promoter element or regulatory element, you need to select the organism in such a way the organisms are phylogenetically not so far a distance where they are present, or it is not too much closer distance they are present. Okay, so what could be the best example? So best example, it will be like you have taken human and you have taken the mouse. So human and mouse, if you are comparing, you will able to understand the promoter and all this aspect, and that aspect you can keep in your mind, and that aspect you can apply for other your vertebrate sequence, and that way you can able to compare. So with whom you are comparing, that is a vital parameter here, and you need to remember, and that way you can easily able to establish the phylogenetic footprinting activity. So the sequence what you are finding and comparing, that is the actual important prime parameter in this case. So these are the again different sites or server is present, and again some of them will be working and maximum of them it is not working. Third method is the expression profiling based method, and this method it is completely depending on that your microarray data analysis process. Okay, so microarray data analysis process, it is called also high throughput screening method, or it is also called the high throughput screening transcriptional profiling analysis method. So here from one particular cell, what are the different RNA is possible to transcribe, all sort of information to be present in a small kind of chip. And my RNA, to whom I wanted to know what kind of promoter was there in the DNA level. So we can compare my RNA or the gene sequence or that particular nucleotide sequence with the chip sequence. In the chip, whatever the mRNAs are there, with all those kind of mRNA, we need to compare. So just imagine billions of mRNAs are there, and your one single mRNA sequence you are comparing with everyone, and how long time or how much time it is going to take, just you remember. It will take huge time, first of all. And second important problem is here, that is during the different conditions of the cell, different it could be different factors which is present in the cell, or that way we can found a different miniature of expression in the eukaryotic as well as the prokaryotic system. For example, if the for bacterial system, if we talk, if it is having like nutrient starving conditions, so some different gene is going to express and some gene is going to suppress, or temperature variations, if we do that time also you can see such kind of variation of the gene expression. And eukaryotic system, if having disease condition, one kind of expression. If it is normal condition, one kind of expression. And how we are telling this is the normal condition, the disease condition, and which there is no particular actual logic. So we can tell this is the normal conditions I have taken that expression of the gene. The animals to whom I am considering this is my control group, we do not know what kind of physiological problem might have in that particular animals. Just I am, for my experimental purpose, I am keeping this is the control group, this is the treated group. Treated group, directly you are giving some kind of influencer. It could be some kind of toxic material, some kind of drug, or etc., some viral infection, bacterial infection you are creating artificially. And that way, what kind of gene expression you are going to get. And the organisms where you have not applied such kind of external cues, and you are thinking that is the control. But in the control also might have some kind of physiological abnormalities from beginning itself. So we are not going to analyze all those parts. So that means the control with the your treated, how we are comparing, that is kind of there is no proper definition, or there is no proper rule. And as we are not looking to the molecular level, but we are analyzing directly to the molecular level, we are predicting directly to the molecular level. So such we are going to get a lot of differences will be there in the actual result, and after doing this kind of expression profile based method analysis. And that's a major difficulty for here. And some gene might be expressed very short length, in the microchips that will be present in the very short length of those mRNA will be there, and some genes will be having very larger length. So in that both the cases, there is a no standard method that if you are the transcript, if it is having 100 or 200 or 500 nucleotide, then only we can able to compare my sequence with that particular transcript. So there is no such kind of clear-cut data also, clear-cut idea also. But that's why I'm telling this is the beginning also I mentioned, this is the not the actual reliable one, only just to study the whether my mRNA what I am talking for that is really present in that particular chip or not. So only that data or that information we can conclude from here. Actually no promoter, no transcription related information basically we can able to get by using this kind of approach. So as it is, I'm talking about the RNA, I'm talking about the your promoter and the regulatory element finding, how it is going to prepare or synthesizing the RNA. So I have put it this word that microarray data analysis could be one kind of process to just to compare, not to give any kind of conclusions. So these are the different process we are following. And again for microarray, this way if you wanted to compare and try to find out by expression profiling based method what are the different sequences are present. So such kind MEM is there, melina is there, inclusivity is there. So again these are all again some of it is working, some of it is not working. And that's why every time I mention that whenever we are tallying any kind of result, or if you're going to publish any kind of result by using the open source server, so you should not rely fully on these. You need to tally your result with the other different server or software, and maximum cases if you are getting the similar result, you can concept that particular result only, or you can consider that particular result only. And here for all the cases PSSM, that is the position specific scoring matrix, is the important criteria. So position specific scoring matrix, suppose we have comparing, these are the particular sequence, of course this is the multiple sequence alignment, and after comparing this way, after putting alignment this way, we are converting this is a multiple sequence alignment, we are converting, or we are trying to find out in each column what is the frequency of each alphabet. That is the first column, whatever is written here, we can see 1, 2, 3 A's are there, and 1 T and 1 C is there. So what is the frequency of each particular column? So that we are going to find out with respect to the each alphabet. And that way we are creating this kind of one table. This is called the frequency distribution table with respect to the each alphabet. And with respect to this frequency distribution table, now we are going to normalize the data. Normalize the data means this is the frequency distribution. Now normalizing the data means what kind of probabilitical concept is there, probabilitical chances is there that A should be present exactly in the first position. So that is called the normalization of the data with respect to the frequency, and that way we are creating such kind of table. This mathematical idea is there, of course. This is, as you know, this is not the correct time to explain the each mathematical process one after another. Here I think already Blossom and PAM matrix, that notes I have provided, so you can go through a bit to get the idea how these calculations are happening. So once you are creating this kind of probability distribution table, now this value we have to take the logarithm by to the base 2. So this log concept we are taking, so this is called the log odd values, and we need to look at these log odd values to understand that whether and how perfectly the sequences are matching, and that way we can tell this is the conserved sequence. Okay, so we just keep in what this kind of table in your mind. And next, suppose this is the sequence I wanted to find out, A, C, T, C, G. Just consider this is the your promoter sequence, might be this sequences I'm looking for from a particular my interest sequence. Okay, so how I can use this particular normalization and the logger score containing table to find out what could be the score of my this particular sequence. In such cases, here whatever the tables we have prepared, okay, so by taking the convert the value of the log 2, so what kind of tables we got from this table, so you can put your sequence AACTG. So with the AACTG, how better way it is going to fit in this particular table. So if you wanted to understand, so we need to look at the each positions, okay, and this position is coming from where? From this particular multiple sequence alignment process. So we can look at the each of the table. So table number 1, maximum value is 1.0. This column maximum value 1.0, and this 3.8. So and fourth is 1.0. 3, why you are taking the 0.8? So as you have seen here, it is having 1.15, because from each table we need to take the value only single time. If it is repeating, then only single time we can consider. So 1.15 if you are considering here, and you can look at their fourth table, it will be the minus value. So minus value we need to omit, and that's why from this table we are considering 0.8, from next table 1, from fifth table 1.38, and from sixth is 1.15. So like that, if we take the summation, so we are getting 6.33. So 6.33 with respect to, you can see here also we have considered ATGC. What could be the probability? What could be the chance to prepare such kind of table? Now, what are the sequence I have provided? That is also made up of ATGC only, right? So this ATGC with respect to one ATGC, whatever the score we are getting. Now this AACTCG, we need to compare. Suppose we need to put it here AACTCG. Now AACTCG, if you are putting with we can found with first position A, so definitely it will be 1.0. Second position is the A. So of course, as it is having 1.0, definitely here also score will be 1.0. Like that, each alphabet wise we try to compare, and finally whatever the score we are getting, if the score is more than 6.33, then that particular sequence we are considering, yes, having conserved sequence, and that's why we can able to consider that is the actual promoter element most probably present. So I'm telling most probably because until unless again we are telling this result with the wet lab result, like footprinting approach I mentioned, with that if you are not telling, we may not able to tell here. But by in silico method, I'm telling you are going to predict only. The prediction is correct or not, that for that you need to go, of course, the wet lab experiment. But for continuing to wet lab experiment, we need to require some kind of data. So the data you are keeping in your mind, and you are continuing your experiment in the wet lab. So that's why this promoter element finding, that is the actual we are completely depending on the wet lab experiment itself. Though in silico method we have tried to optimize it, but finally to predict, yes, it is the only things. So we need to depending on your wet lab experiment. So I'll be showing here how basically we are going to find out it by using the different software tools. So for that, I'm going to tell you the finding the promoter sequence and transcriptional factor binding site, how practically we can execute it. So if kindly you open your laptop, if you are using, or desktop, if you are sitting with that, kindly follow me what exactly you have to do for finding the promoter sequence and the transcriptional factor binding site. So for example, I have taken the sequence of Homo sapiens. So here you need to remember, you need to, for this particular first experimental process, whatever I'm showing, so you require the mRNA sequence. Here, if you are using the DNA sequence, you are not going to get the result. This process you are not going to follow. Okay. So here you need to remember, you require the mRNA sequence, because with respect to the mRNA sequence, you are going to analyze the your actual transcriptional factor regulatory site as well as the promoter site where it is present. Okay. So first experiment. So you are opening the NCBI and you are opening the one particular sequence. So I have taken here Homo sapiens interferon lambda 1 IFNL1 of mRNA sequence. So you can see here. If you are opening the this GenBank, that particular sequence, so first will be definition line, organism, experiment, everything will be mentioned. And if we scroll down, you can see the source of the organism and the gene, exactly from which position to which position perfectly the gene sequence is present, and what are the exons are present. Then CDS, that means that is actually protein coding region. Okay, so exactly it is starting from 16 until 618 it is there. That means starting position, your codon is 16 alphabetic position, it is present in the sequence, and the stopping codon is exactly present 618 positions. And with respect to that gene, again, that what kind of protein it can able to encode, that also it is mentioning here. So all these things you need to remember if you are opening the NCBI, and if you are opening any particular mRNA sequence, if you are using any kind of mRNA sequence, please should not close down that particular window. That is, you need to require after some times also, and you will see where you are going to use that information again. Okay, so kindly do not close that particular window once you are opening and using this particular sequence. Fine. So you got the one kind of sequence. So I mentioned you need to look at the exon part, you need to look at the CDS part. So all the things you need to keenly observe and you have to note it down. So these are the things, like it is having a lot of exons. So wherever exactly the exon positions are there, and all the things they are highlighted here, this position to this position is the exon. It is already mentioned along with the particular gene sequence of or we can tell mRNA sequence. And lastly, the gene wise the alphabet have provided from where this mRNA sequences has come. Okay, so all those things you can look at after opening that particular server or software. And even you can see some regulatory part elements, exactly where it's present, poly A site, exactly which position it is present. So everything will be mentioned in the GenBank format. Okay, so I have taken IFNL1, and now you required for analysis purposes the FASTA format of this particular mRNA sequence. Okay, so you just click on the FASTA. If we look at the first part when you are opening the NCBI, so if you click on this FASTA, you are going to get the sequence of mRNA in the FASTA format. So I got the sequence of mRNA and I pasted over here in one Word file, otherwise one notepad. So after pasting, I got this is the sequence. Fine. And you just remove that greater than symbol and whatever is written, you just remove of it. Why you are going to remove of it? Because you are going to find out that upstream regulatory part, whatever is there. That's why you are deleting the definition line and just keeping the name of this particular mRNA. So here IFNL1. So the name I have kept, and after that I've given the gap, and you can keep or you can paste that particular sequence whatever you have taken in the normal text format. I'm telling normal text format, no FASTA format here. Okay, so I have taken this. Fine. Now what you need to do, you need to open your this particular in the Google, you need to type eukaryotic promoter database. I repeat, eukaryotic promoter database. You are typing, and you are whatever the homepage is appearing, you just click on that EPD, the eukaryotic promoter database. So you just click on that, and it will be appearing that homepage this way. Okay, you can see, look at here, EPD. This is the Swiss Bioinformatics Institute. From there, it has it is taken care. So they have designed it. So after opening it, what you need to do? You need to. These are the two different boxes you can see. So this is one box, and second, this is, it is mentioned another box. Fine. So you need to type something here. What you need to type here? So the gene, whatever the sequence, whatever you have taken, I have taken here IFNL1. So I am typing here IFNL1, and if you click here, you can look at the particular arrow symbol. So if you click on this arrow, here a lot of options will come, whether Homo sapiens or all the animals name, vertebrates, invertebrates, everything will be mentioned here. And as you are going to analyze with the Homo sapiens gene, what kind of promoter and the regulatory part is present. So you select here H sapiens, that is the Homo sapiens. Okay. After that, you need to just click on the search. If you are clicking on the search button, immediately this page is going to come. I repeat what so far what I have mentioned. So you are opening the NCBI, you are taking any gene mRNA sequence, you are going through what are the exons, introns, all those aspect is there. You just going through of it, and the sequence in the FASTA format you are copying in the text file, otherwise Word file. Okay. Next, what you are doing? Next, you are typing in the Google eukaryotic promoter database, EPD. So you are clicking on the EPD, you are getting the home page. In the two boxes are here. So first box, which gene you have taken, mRNA sequence you have taken, we write down that, and in the sequence mRNA, it is present in the human, present in the plant, or present in the so and so. So you need to specify that. As I have taken the human, so after clicking here, I have chosen the Homo sapiens. Okay, and then you are clicking on the search button. So once you are clicking on the search button, this is the result page. It is appearing in front of you. Okay, so first you look at the result page. This is the top part. Okay, so top part, it is written something. What is written? I am coming, and then some graphical presentation is there. And so this graphical presentation, if you scroll down, so further you can see it is better way, and then completely again if you are scrolling down, almost at the end of this page, this is the information given. Okay, almost end of the page, this is the sequence retrieval tool, and something is given. So in the result page, if you look at the first part, so here what is given, it is mentioned here the sequence of the mRNA, what gene I have provided, the name is mentioning here, and the promoter type, the single promoter they have found here with this particular sequence, and then organism. So it is the Homo sapiens, already have selected Homo sapiens, gene symbol IFNL.1, this is a gene bank symbol, and then the sequence, look at here, I've highlighted here. Okay, the sequence, if we enlarge it, yeah, I think now it will be much clear. So if you look at the sequence, so look at here few alphabets are mentioned in the capital, whereas the starting few or maximum word or alphabet is mentioned in the small letter. So this is the important actually. This is the small letter and this is your capital letter. So what is the meaning of it? The small letter and the capital letter. So this is only you need to understand better way here. So the small letter and the capital letter word, the small notice here the small and the capital alphabet are mentioned. The small letter indicates the promoter regions. Fine. and the capital letter indicates the transcript region which is already there in the mRNA sequence and which you have used in the FASTA format. Got it? So here you are getting the small alphabet as well as you're getting the capital alphabet, right? The small letter alphabet, it is indicating that is the promoter regions, and capital letter, these alphabets are indicating these are the already part I have provided or you have provided along with your sequence, and that is indicating the transcript region. That means mRNA, fine, mRNA region that is. Got my point? So the small letter and capital letter, it is indicating like this way. And in this graphical picture, if you look, so wherever this blue part, okay, this blue portion it is there, okay, the blue cone, this symbol wherever it is there, so that is indicating that is the actual part where the polymerase enzyme will be bind. That means this is the actual promoter site most probably present with respect to my promoter. So this is the graphical presentation. Fine, this blue part is indicating, I mentioned here, the blue indicates maybe a promoter region. Got it? And further, if you scroll down, you can need to look the sequence retrieval tool. So here in the down of it, it is written the sequence retrieval tool. In the sequence retrieval tool, with respect to the IFN, it is mentioned sequence IFNL from -449 to 100 it is mentioned, okay, and further below the search motif tool that is mentioned here from 1000 to 100. Got it? From 1000 to 100, and here it is mentioned the sequence retrieval tool with respect to my sequence -499 to 100. So what does it is indicating? Minus, I mentioned that we are having conventional approach. The starting positions of transcription will be +1, and whatever the sequences are present in the left hand side, that means upstream part alphabet we denote by the minus symbol. Got it? And then this minus symbol will be continue till to the 0, and from +1 it is going to indicate exactly from that position transcription is going to start. Correct? So this is already all of you know. So now look at this analysis here. Here, remember total length of the promoter sequence plus 5' untranslated region sequence always to be 0. I just now mentioned. So whatever the minus, whatever is there, the minus part as well as the 5' untranslated region together, conventionally concept or we can accept that is the till that much position is the 0. Got it? So here you just look at the 0. So here what we are going to do, so the 0 why we are telling, I hope you got understand. So now here -449 already mentioned here, -449 plus 15 we need to add. From where you are getting this 15? For understanding this 15, you go back to the gene sequence of NCBI, where the series is mentioned. Here in this case of the sequence, the series is starting from 16 to 618. That means before that 15 alphabets are there. Okay, so if we consider after 0, 1 to 15 alphabets are there. After 15 alphabet, thus from 16 onward, the transcriptional or protein synthesis process actually start till 618. That's why I'm telling before that, after 0 will be having another 15 alphabet. That's why we need to add here this 15 here. Okay, plus 15. So plus 15, 490 minus 499 plus 415, you are getting -484. Correct? If you are joining is plus minus minus, and it will be -484. And how long this particular untranslated means upstream region will be there? Upstream region will be there as per the software. They have predicted the motif site from -1000 to +100. Okay, that is the region, and we are considering the minus positions because we are only looking this motif is present to the upstream region. That's why that number whatever we got minus 484, we need to add it with this minus 1000. Got it? So we are getting minus 1484. So minus 1484, you got the idea. Now you need to get the sequence with respect to the mRNA whatever you have provided. This total 1484 alphabet in the upstream region. I hope you got the idea. In the upstream region, that regulatory sites are present along with the promoter. So now we are looking that upstream region here. Fine. So we need to change this alphabet here right now. Instead of minus 44 minus 499. So you just check, you just change it, this minus 499. You just write down this alphabet minus 1484 to 0. Okay, instead of 100, you need to make it 0. And then you need to click on get sequence. Once you are clicking on the get sequence, immediately this sequence you are going to get. Like, okay, so this sequence you will be getting, and you need to copy the sequence, and the sequence you need to paste here from the red whatever is written. From here, this red alphabet, it is starting actual my mRNA sequence. From here to actually this much is my mRNA sequence. Already I have taken from NCBI in the FASTA format. I have pasted beforehand. Got it? And these are the sequence, these are the part I have calculated from here. And after that, I click on the get sequence. I got the sequence here. I have copied that and I have pasted just before of my sequence. So these are the sequences indicating the total regulatory region portions where the regulatory elements can bind and ultimately can control the promoter activity. I repeat, this is the sequence whatever you got that should be present in the DNA level along with this particular sequence. And DNA level, this sequence is definitely promoter also to be present and regulatory all element to be present. And for transcribing this IFN-L1, obviously in the genome level, this particular positions present. That's why this gene is going to express. I hope the idea is clear. Now, another important things I mentioned here, you just go back to the NCBI sequence and you can found here 16 to 618 it is mentioned. That means before 15 alphabets are there. Correct? Now, if we come to this particular original sequence, repeat, if we come to this particular original sequence and if we count the alphabet from the starting 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, right? Exactly after 15, from 16 onward, we got the starting codon. Got it? ATG is the starting codon. Am I right? So here also it is mentioned the series is starting from the 16 positions. Look at here, series is starting from the 16. That means my sequence exactly from the 16 position, 16, 17, 18, these three together is going to giving the starting codon. And before that, whatever the portion is present in this sequence, that is called the 5' untranslated region. So these 15 alphabets are the 5' untranslated region, and that's why I have highlighted each. And ATG, I have highlighted with the green because this is the starting region. Okay, from here actually protein is going to start, and that information only is given to the NCBI. And same informations we are applying to analyzing along with this untranslated region, whatever the other regulatory region should be present to express this kind of mRNA and to produce the protein. Got it? I hope it is clear. So I mentioned here that 15 alphabet is the untranslated region. I gave this explanation. So this all the upstream region, whatever the portion is there, plus 5' untranslated region, it's together is considered as the 0 as per the normal convention of molecular biology. So this is the 0, and that's why in the software we have mentioned -49 499 plus 15, this we need to add, and then the prediction cutoff is 1200. So from 1000, that is the -1000. So this 1000 we are adding with that. So it is coming total -1484. And this 1484 region to the upstream, how much sequence is present? So for that we are changing it -1484 to 0, and we are getting the get sequence. We are clicking the sequence whatever you are getting. So this sequence completely is indicating this is the my all upstream region sequence. So here the TFBS, Tata factor binding site, and all the site will be present here. And this is the red part, this is the untranslated region part, and ATG is the starting part. Got it? Now, as the concept we are having, so -10 or -35 region, or especially yeah, -35 region, if you wanted to find out exactly the Tata part if it is present or not. So now this is the starting part, this is the upstream. If we go back, definitely some here if you are reaching to the -35 region, so definitely you will be getting some kind of consensus alphabet, and that consensus alphabet is your promoter region. And eukaryotic system, I explained how we are predicting it. So together the upstream region plus promoter region we are finding out, and from that particular region, based on the concept of this promoter sequence, we need to get the idea exactly where your these particular promoter elements are present. So if you count it from here, if you go to the -35 positions, you can see at least 4 alphabets will be common with the normal conventional eukaryotic sequence, this promoter. Okay, so this is the one way. So we got the entire sequence with respect to the gene, what is the untranslated region, where exactly the total regulatory sequence along with the promoter part is present. Now, further, if you wanted to analyze it, verify it, what you need to do, you need to type in the Google again, promo TFBS. Okay, PROMO TFBS. So if you are typing and opening, you are going to get, this is the home page, promo homepage. In this promo homepage, once you are opening, you can see here some hyperlink conditions are provided. So select species, select factors, etc., etc. So you need to click on the select species. Okay, the first option you need to click on select species. You are going to get, this is the page as you have clicked on the select species. Got it? Now, as my sequence is the Homo sapiens sequence, so here the options whatever is given, I need to click on this option. So first option, like considering factor, so only the human factor I am clicking. Considering the sites, so only the human sites only I am clicking. Got it? Because my sequence is the human sequence. And if you're having doubt with the sequence, you go for all factor and the all sites. Got it? As I am definite and confirmed, this is the human sequence. So I am selecting only the human factor and the human sites. Actually, what I'm looking here, I'm trying to find out what are the various transcriptional factors can bind with this particular selected or this upstream region with respect to that IFNL1 mRNA sequence. Got it? So I wanted to find out what are the various transcriptional factor having tendency to bind in this particular upstream translated region. Got it? So for that, I have selected only the human. After selecting it, you just click on the submit. So after clicking on the submit, you are going to get again this particular page. After clicking on the submit, this is again repetition. You are going to get, this is the particular page. This is the repetition happened. Okay, don't worry. So after this particular submit, you are going to get immediately. This is the page, search promoter site, and here the sequence you have to paste. Okay, so what sequence you need to paste? Click on the search site and the paste obtain promoter sequence from the previous experiment, and then you submit. So previous experiment, so we got, this is the entire upstream region signal sequence. So you just need to copy entire thing. Okay, so till this much you need to copy and you need to paste over there. You need to paste it here. You need to paste here. And of course, no FASTA format, nothing is required. Just sequence you copy and paste, and then you click on the submit. Once you are submitting, this is the result page final you are getting. And you can see here, this is the upstream region sequence. And 1 to 10, 10 to 20, 30, 40, 50, like that it is given. So if you are scrolling right side, and if you are scrolling right side, you can see that till 0 all the total nucleotide whatever is present is mentioned. And second thing, this color is indicating the corresponding transcriptional factor, what can able to bind exactly which particular nucleotide sequence. So if you look at the 6, you can tally the color of here, whatever the yellow color is there. 26 is having yellow, 66 is having yellow color, 6 is having 46 is having. If you scroll it right side, many more yellow colors you are going to get. So the all yellow color, this transcriptional factor, whatever is mentioned in this table, it may have, all these are having the tendency to bind in the respective positions of this particular sequence. And of course, the 6 and 26, this number also it is going to indicate. If you scroll it right side, the 6th position, definitely what transcriptional factor is there, and that transcriptional factor is going to bind between 60 to 70. So that's why the 6, the number also mentioned here. Like if you look at the 26, you can look at here 26, that means it is having tendency to bind within 90 to 100 this particular region. So that way you can analyze what are the various transcriptional factor having tendency to bind with this particular upstream region of your analyzed gene sequence, and that way you are going to predict, complete your result. You can able to write the information. So for expressing so and so gene, this could be the promoter element, this could be the total upstream region sequence, and these are the following transcription factor can able to bind along with my these genes what upstream region. And you can, this is useful very much to understand the cancer research. So some paper, if it is mentioning so and so gene expressing because of so and so transcriptional factor, and its overexpression, it is going to do so and so biological activity or biological consequences. So you can understand that sequence, this particular promoter exactly where it is binding. So you can able to make your plan so that this transcriptional factor may not able to bind over there. And if you're able to make that, so the cancer research, you have some particular problem you can able to solve. So this is all about that how we can find out the entire transcriptional factor binding site with respect to the mRNA sequence. And next part, directly if you wanted to just finding out the promoter part, so that also we are having server, that is promoter 2. Okay, or you can just type promoter, you are getting so many things. You are just click on the promoter 20. So you are going to get the home page, and here just you paste your FASTA format sequence. Okay, so FASTA format sequence, of course, the DNA sequence you need to paste over here. No need to paste the mRNA sequence, no need to use the mRNA sequence. You can paste the DNA sequence over here in the FASTA format, and then you can just click on the, after clicking on the full output, you need just clicking on the submit. Okay, so after like I have taken the adenovirus type 2 complete genome for this experiment, and I have pasted it in the FASTA format. I have clicked on the full output and clicked on the submit button. So once I am clicking on the submit button, getting this kind of result page. So initially first the sequence information only it will be written, and each line will be having 60 alphabet. And further if you scroll down, you will be getting the analysis part, where it is written the positions, that means 600 positions. They have predicted here highly likely prediction of the promoter sequence. At 1800 positions also they have highly likely predicted. So like that, here the software is going to analyze the each and every possible positions of the promoter, and the respective score also they have provided, and based on the score they have mentioned highly likely prediction or moderately prediction, etc. Okay, so you need to understand what is the meaning of highly likely prediction, what is the meaning of marginal prediction, etc. So as we've already discussed that for prokaryotic site is having -10 region, this is the sequence, the TAT sequence, and site 2 that is the -35 is having TAGHCA sequence. And similarly in the eukaryotic site we can found almost -25 region TATA and -75 region, which is called GC box or CAT box, CAAT, the sequence will be present. So software will be try to analyze if you are providing the prokaryotic sequence by keeping these two alphabet or these strings of stage of alphabet, they'll be keeping mind, and they'll be trying to find out in your provided sequence wherever these particular sequences are coming. If you are providing the eukaryotic gene sequence, so these particular positions, whatever the sequences are present, so that software is going to study those, and finally this way they are going to give the result. Of course the score is the statistical score, and this statistical score is having significance. Like the score, if you are getting below 0.5, we can ignore that part. That means that is the not good predicted result. So if we found here in between anywhere less than the result 0.5, we can omit that. So never ever we are going to consider, suppose say for example here 0.538 marginal prediction. So we can omit this particular site because as per this rule, it is not fulfilling perfectly. So a lot of 0.565, 0.538 they have predicted, like here also is having 0.56. So these are the positions we have to omit. We are not going to consider that part. Like the score, if it is coming 0.5 within 0.8, so that is going to tell you a marginal prediction. So marginal prediction means almost 65% of the true transcriptional start site out of 100 base pair of the upstream software able to determine. So we can select this particular part, and it will be written marginal prediction. So you can see here written marginal prediction, many things. I think I can zoom it. So you can look at here marginal prediction mentioned, highly likely prediction mentioned. Okay. So these are the score is indicating that, and if the score is coming 0.5 to 0.8, this is a marginal prediction. And if the score is lying between 1.0 or more than 1.0, that is the highly likely prediction. That is 95% confidently the software has predicted this particular part, and definitely we can use those parts. Like 600 it is mentioned highly likely prediction. 1800 that particular position score is 1.178 highly likely prediction. So if you wanted to analyze this 600 position, which particular sequence is present, so you just go back here. 600 means you can see here this is the 600. So you can just look at here in the 600, the first alphabet is the A, and then you can come to the next line AATGGC. Okay, so this is the once again conserved sequence, and that is the promoter sequence, and that's why the software have identified with the highly likelihood prediction process with respect to this score. Like if you wanted to again find out this highly likely prediction at the 6,200 positions, so you can go back. Already I have not taken that much screenshot. So if you in between, if you look, so if you are taking that 1,800 positions highly likely prediction, so 1,800, again I have not taken the screenshot. So anyhow, whatever the result here it is given. Okay, if you wanted to tally, you come back to the 600 position. That's why I mentioned here every line will be having 60 alphabet, and if you scroll go down, so 60 plus 60 plus 60, like that it will move. So exactly which position it has mentioned, it exactly count that particular alphabet, and from that particular alphabet onward, you can find out that original sequence as per the rule of the prokaryotic or the eukaryotic system, it is there or not. So this way we can use directly to finding the promoter element, promoter sequence. But I repeat, it is not going to tell you how much that transcriptional factor binding site, the length of the transcriptional binding site, total length of the gene, you are not able to predict from here. Only the promoter site you can able to predict from here. Next, third one, and this is the last one, the BPROM of softberry. You can type, this is the URL of things, you can type on the Google, and then you are going to get, this is the home page. You can click on the left hand side, there is a search for promoter and the functional motif. You just click on that, and then you are going to paste your sequence in the FASTA format. Okay, so here remember, you need to provide the complete genome sequence. Partial sequence if you are providing as per this cutoff value or the statistical parameter, sometimes may show that no starting site predicted or no promoter site predicted in this particular provided cutoff values. Okay, so you need to remember the entire genome sequence, lot of organisms entire genome sequence available. So those sequence in the FASTA format you need to provide here, and leave it by everything's default. Okay, and if you wanted to check the only the Tata box, so we just click on here Tata, otherwise just leave it and just choose the, you can just go for the process, this option, and immediately that you are going to get, this is the result. Fine. And what does it is indicating? I just zoom it. So it is going to indicate you that so first thing is the promoter position, it is mentioned. They have mentioned this is the what sequence and everything, total length of the sequence they have mentioned, and the 3 promoter or enhancer part they have predicted. Okay, position is 1853, and this is exact positions, and this including the enhancer part they have predicted, and exact the sequence of the promoter, that is the TATAGTG, it is present 1823 from that particular positions. But rest of that, whatever the alphabet is what are the other number has added here, that is indicating your enhancer positions is present. Like the second prediction they made at enhancer part 875 these positions, and you can look at the Tata box. This is the sequence is present at 843 positions. And look at here that with respect to the Tata box, they have predicted it. And with respect to the Tata, these are the statistical value they have set up. And based on that, they got this result. Okay. So this way also you are going to understand with respect to that provided entire genome sequence. So where exactly that Tata box part is present, and with respect to the Tata box, so what are the different enhancer position also it is there. And last part, that is a bacterial promoter, and for the bacterial promoter also the software is the same site. So if you go to the top, you can find the open on gene finding in bacteria. You just click on that, you are getting the BPPROM, this server, and you are pasting your sequence in the FASTA format, of course the DNA sequence, and you are just clicking on the run. So once you are providing the sequence and clicking on the process, you are getting again, these are the similar kind of result. Here basically they have based on the promoter by operon part they have selected, fine, which sequence have given, how many number of nucleotide is present they have mentioned. So threshold parameter they have mentioned here 0.20, and number of predicted promoter they found in the only one promoter they found with respect to this threshold value. So minus 10 box, at the position they found 395, and the score they got 44, it is more than threshold score, and minus 35 they this particular box exactly at present to the 378 from this particular position, and the sequence is like that, and the score is 55. Okay, so this way we can able to understand that for the particular promoter where exactly and what is the sequence exactly it is present with respect to the prokaryotic gene. Okay, so oligonucleotide from known TF binding site, no Tata factor binding site they did not found, because the sequence whatever you have provided, which hot stage of the sequence and the threshold value is important here, and no such site for promoter at the 410. And in the among the total 771 nucleotide, so 410 nucleotide, they have not found any kind of such kind Tata box or this box is present. So this way also we can able to predict the promoter element where and which position exactly present, how much it is present. So with that, thank you very much. With that, I'm completing my session. So please, if you are having any queries, kindly ask the question. Thank you.
Hello.
Yes.
In case of software, you have shown the results
Uh-huh.
where some numbers are there, then dots are there. What is the meaning of that?
Number are there, then dots are there. Which one you have mentioned? Software.
Software. Before
Okay.
bacterial promoter.
Okay.
Yeah,
This is your talking, sir.
Yeah. LDF, then plus 13, then dot 682.
Sir, these are the statistical probabilities value.
Okay.
This is a statistical probabilities value.
Data box it is at for first case 1823.
Yeah. Yes.
And then it is also statistical value plus 4 points.
Oh, of course.
Yeah.
Of course.
Another question is in case of eukaryotic promoter database,
Hmm.
in case of result, some colors are there. That is for initiation codon, it is in green, then promoter region is red. Is it automatically comes?
No. For make you people understand, I made this one. Also, in my sequence you are talking, right?
Yeah.
No, for make you people understand, I made it. You also can do similar way. Otherwise, you will not able to understand that how many alphabets are present just before the starting codon. So this particular part is the untranslated region part. So make you people understand, I manually I did this.
No, it is your sequence, but in case of results section, you have also colored like this.
Yes, because this is the sequence. Now this is the sequence I am having. I understood this is the untranslated region, and what I did, whatever the sequence I got, like minus 1400 something, I just paste it before this. I kept the cursor here and I just paste those sequence.
Okay.
Because upstream region, it will be there before the untranslated region. So I just pasted over there, nothing else.
Okay. Thank you.
So you will be able to understand exactly this is a sixth position,
Yeah.
what is ATG is there.
Wow.
For that only.
Thank you.
Welcome. Anything else from anyone?
Dear participants, any questions?
I think they do not have any questions, sir.
No,
You
sir.
can continue your session, sir.
Somebody is asking.
Yes, please. Hello.
Hello. Somebody.
Yes.
Somebody. Hello.
No, sir. No one asking.
Today we will do with the molecular docking studies. So I have shared the link to download the PYRX software. PYRX in short it is called as. And there are two versions within that. One is I will share my screen. So one is commercial version and another one is free version. So this is the link that I have shared yesterday. Why is internet is? How about you? I hope you are able to go to that page, right? Here in my college, it is not allowing, but nevertheless, I hope for you it worked.
Yes, sir.
Could download and install, right?
Yes, sir. I have downloaded.
Okay. I hope others also could download and install the tool. Okay. Now, as we started discussing the concept of molecular docking, right? Yesterday, I have given the example of lock and key model, right? And how should we go about the docking? In the sense, the principle and structure-based drug designing, ligand-based drug designing, and from where we can take the ligands, right? And from where we can take the protein. So we looked into PDB, right? We downloaded the structure, and we also looked into how we can remove B chain, retain only A chain, and zinc database for getting the ligands, and then other databases servers like ForMapper, Campbell. So those things we have discussed in our previous class. Now, based on all the understanding that we did in our yesterday's class, now we will go for performing the molecular docking. Now, again, as I mentioned in my previous class, the molecular docking is not like the first step, right? We got to do the pre-processing of the files. That comes under the design of experiments. Now, we want to do the docking of the ligands. Today we will do with the protein ligand docking. Maybe later we will do with the protein docking, because we use the protein ligand docking widely. And to do that, we need this PYRX software, right? That you have installed already. We went to this PDB, right? Then we looked into the 4MBS structure, and when we type like this 4MBS, then we know that we obtained this crystal structure of the CCR5 protein. Along with this, we have this Maraviroc ligand bound to it. Now, what we will do today is to simplify the concepts. Maybe I will just write a bit about the procedure that we follow. Okay, over here, I will use a pen, maybe color. Okay. Now, what we will do is, let me take this. Just understand the concept again. What we have in the 4MBS, okay, so you can also make a note in your book, because I may need to delete this page later. Now understand the design of experiments, okay, because when you want to do a docking, this becomes an important aspect. Now 4MBS is nothing but our human CCR5. Now in this 4MBS, we have two components present within this. One is the atom section, that is the protein section. Let's say protein, I will write it like PROT. Then we have the ligand which is bound to it, that is the MRV, that is the Maravirok, right? So we have the ligand and we also have a protein, and this acts like our complex. So this is the complex that we are downloading from the PDB. Now, this is the complex now. Let me put this one in bracket. Okay, now this is the complex wherein we have the CCR5 protein, and also we have the drug or nothing but the Maravirok that is bound to it. Now, what we want to do is docking. Now, do we have the energy associated between the CCR5 protein and the Maravirok available? When I say the binding energy, right? So based on the program that we use, so we will be using Autodoc Vina. Okay. So in few cases, we will refer it as a Vina score. If we use the gold, we call it like a gold score. That is from the Schrodinger. So most of the time in when we do the molecular docking, okay, so the reviewers won't prefer us to use the term binding energy, because we get the binding energy when we do the molecular dynamic simulation. So when we do with the docking, it is preferred to refer it as score, okay, Vina score. Now, what we are looking at, again, if I come back to the core aspect, we have a protein, we have a ligand, we do not have the Vina score available for this complex when we use Autodoc. Autodoc is the program, or you can think that as a kind of an algorithm. So just like in the today's presentation or even in previous presentation from the professor, it was mentioned like when we are doing, right? So it was mentioned like hidden Markov model and hidden Markov model algorithm, position specific scoring matrix, PSSM, the PSSM score, right? So for each activity that we do, we will have the corresponding score or the value available. So in the same context, whenever we do the docking, we want the score. Since for this complex we do not have a score, and our aim is to develop another drug molecule which acts much better than the Maravirok, right? We need to have a score as our standard. And how will be the score usually, right? Just like we obtained the score even when we did the BLAST, right? Bit score. When we did the FASTA, we obtained the score. Even when we did with the pairwise alignment or multiple sequence alignment, there also we got the score. So in the same way, whenever we perform this, we need the energy or we need the score. So the score will be something just as a representative, maybe like -7. The unit for this will be like kilocalories per mole. Okay, so -7 kilocalories per mole. So this will be our unit. So -7, -8, -9, -10, okay, -5. Even it can go like -7.2, -7.3. Even we can get that one in the decimal form. Now this is how the usually score will be. Now let us assume, okay, we will do this experiment, how we can obtain the energy, okay, that we will do now. But along with this, right, so what is the purpose of getting this score? The purpose of getting this score is to get the existing drug, how it is interacting and how much energy it needs, okay, for the existing drug, which is the Maravirok, which is there in the phase 4, that is in the market. What is the energy that it is going to take to associate with this particular protein. Now, if the energy, for example, as I mentioned, if it is like minus 8 kilocalories per mole, and you want to develop a competitive inhibitor, so how is the energy or how is the score for your molecule? So if the designed molecule gives like minus 9 kilocalories per mole, then we say like this is much better than this. Okay, so the minus 8 and minus 9, if we compare, since the energy is in negative, okay, lesser the energy, better it is. So the minus 9 is better than minus 8. Okay, then we also look into the number of hydrogen bonds, interaction, all those things. So first thing you got to remember is this score. Since we do not have the score, we got to perform the docking for that existing drug and get the score associated. It is as simple as just to put the entire stuff for the easy understanding. Most of you would have done the gel electrophoresis, right? Now, when you are running the gel, you will also take something as a marker. Is it like the molecular weight and other stuffs for the marker is not known? It is known. But then why are you keeping it when you are running the gel? So it is just to know whether the gel that you have prepared, when you know the marker and where it should stop. If the marker is also going to stop at the same place, then you know that the preparation is correct. Okay, so you know that the preparation is correct. So you know where the band should stop. And when you run this particular experiment, if the band stops at the exact place where you have the corresponding molecular weight, then the preparation, the movement of the gel, everything is perfect. Then for the unknown, whatever you have, right? These becomes like our valid results. What if you do not have anything like a reference? Then the question comes like whether whatever the molecular weight we are getting for this band is correct or not, because you do not know anything to take it as a reference. Are you getting? So because of this understanding to happen, now the Maravirok is same like the marker for which you know the information. So just like the marker, we are using the Maravirok as our reference. Since we do not have the value associated, bending assignment, bending value or the score associated, we will run this ligand along with the ligands that you are interested to check. Okay, you will check other ligands, whatever you may have it, like 4 ligands, 5 ligands, 500 ligands, 50 ligands, right, or 70,000 ligands. In one of our project, we did up to 70,000 ligands already. Now we are interested in doing with 3 lakh compounds. Okay, so it's quite possible. You can take n number of ligands and keep it for the simulation or keep it for the docking to happen. Okay, that is not a problem. Now, I hope this particular concept is clear, or any doubts with this?
Sir, we have to run the gel along with the protein as well as the
No, this is correct, but see, this is just to give you the analogy. We will not do this one. This is just to give you why are we taking this MRV? Okay, why are we taking this Maravirok?
as a standard.
It is, yeah, as a standard. Just like we use standard in our gel electrophoresis, in the same principle, we will use this as our standard. Then we will have multiple unknowns. So here also we will take multiple unknown ligands. Unknown ligands means we do not know whether it is going to bind to this protein or not, that we want to test. These are the test ligands. This is a known drug molecule. Now, when we know how is the interaction, when we do the docking, are we getting the same kind of result, whatever the crystallographic result is giving, or if it is a deviation, how much is the deviation? If the Maravi rock is going to interact, now again, to put this one into a simple understanding, what we are doing first is we are doing the tool validation. Okay, we are doing multiple things. One is like tool validation. What is that tool validation refers to, right? Whether the algorithm is working fine or not, whether the docking is happening correctly or not. So if you can remember the experiment or the example that I have given, when you purchase a calculator, right, what we do is, first thing we do is, we will not do something like sin x and function apply that this. No, we will not straight away do the execution of any of the calculus or any of the calculation. First, we start with the simplest method, that is 2 plus 2, whether we are getting 4 or not. Then do with the 2 into 3, are we getting 6 or not. So you will just quickly check whether the calculator is working fine or not. Once you feel yourself comfortable, yes, it is giving the result. Now what you are doing, what you did there, you know the answer already, right? It is not like you have performed like 1068 into 1056, then check what the answer is, because we do not know the answer for that. So that those things we will not do. We will do with 10 into 10. Are we getting 100 or not? If we get the answer, whatever we know, and if it matches, then we will get the confidence that yes, the calculator is working fine. Now the tool validation refers to, we know how is the Maravi is interacting with the protein. Do we know how is the interaction of the Maravi with the protein? Anyone?
Yes, it is known.
Yes, from where we got the result, from where we observed the interaction?
From PDB.
Yes. So from the PDB sum, right? So when we performed the PDB sum yesterday, so we know this one. We got already MRP, the Maravirag, how is the interaction, how is the hydrogen bonding, right? We know this is the standard. And if we do the docking, this is something like 2 plus 2, 4, we know that. Now what we want is, in place of calculator, we are having the software, that is Autodock. Now when I run the same protein and same ligand and run in the software, just like when you type and run in the calculator, it gives the result as 4. When I run, whether I am going to get the same result like 2 hydrogen bonds or not. If I get the exact thing, exact result, then I will have like 100% confidence in the tool that yes, it is. But what if we get like 1 hydrogen bond, right? Then we will say like it is accurate, okay, but since it is a calculation, right? So yes, it is somehow giving the nearby results. Okay, that is how our interpretation, because there is a difference between the calculator and the tool that we are going to use. So we want 2 plus 2 is equal to 4, but what if it is given like 3.9999 something? Then we say like, if there is no other alternative to use any other calculator, that is the only option if we have, right? Then we say okay, it's almost nearby value what we are getting. Okay, that is how our interpretation will go to. Now our design of experiments, if you think of, what we need to do is, whenever we want to do a docking, we need to have one file for the protein and one file, separate file for the ligand. Now what we have is a complex. For MBS as one complex file I have. Now what we have to do is, we got to separate the file. One should be referred as protein or 4MBS A chain.pdb. Okay, that should be our one file, and another file should be like mrv dot mrv.pdb as the second file, that is the ligand file. Then if we want to take some more ligands, we can take those ligands, and again those things should be in PDB. If it is not in a PDB, we have OpenBabel as a server, also a program that is available within the PYRX to convert it into a PDB. Okay, that we will do now. Then we can take all these ligands, allow these ligands, okay, allow all these ligands to interact with this protein. We will allow all these ligands to interact with this protein, then check what is its Vena score is. If the MRV is having like -9, L1 first ligand is having -8, second ligand -7.2, third ligand -7, fourth ligand, let us say -10, for example. Okay, then we will get to know like the Meraviroc is having -9, but our ligand is having -10. So L4 is better than any other ligands. That is the first information from the values or from the score. But we can also check with the hydrogen bond, the interaction, where it is. Then you will decide which one. With the time, I will just explain how you can decide which one is better. Okay. So any doubts with whatever we discussed so far in this?
No, sir, clear.
Okay. Thank you. Now, can you tell me how we can separate it out, the MRV from the protein which is there in this file? How can we separate the MRV from the protein?
From raw file, we can take the data. We can delete the MRV portion.
Correct. Using, how can we remove, like
by selecting and deleting, then we can
yes,
visualize it in PyMol.
yes, perfect. Now what we will do is, okay, so we are here. What I will do is we will do from the scratch. Okay, so you might have created a folder like the new college Chennai or workshop or whatever. So create one more folder within this called like docking. Okay, and we do not have anything in this particular folder, so you can also do the same thing. You create with the name of your comfort, docking, workshop, right, whatever. Then this is blank. First thing we need is to download the PDB. Okay, download this PDB file. Then save this one under this docking file, docking folder. Okay, so since we already have 4mbs yesterday, that is why. So 4mbs.pdb, you save this. Now in this particular folder, we have one file called 4mbs.pdb. Now you can open this in the PyMol, okay, or another way is open this file in the Notepad++, okay, and then within this Notepad++, we got to remove the B chain, and we got to remove the entire B chain, and we know the difficulty that we did yesterday. So if we have that particular file, okay, only A chain, this is what, if you have this particular file, you can use this. This is only with the A chain. But if you do not have this, assuming that those files are not there with you, okay, I will use it as if we are doing for the first time. Okay, so don't consider anything whatever we did earlier. Right click this, open in a Notepad++. We will have this flat file. Then scroll down, delete B chain, you know that after this TR termination, this is our B chain. Select the B chain. You can hold the shift and then use the downward arrow key to select. That becomes easy rather than scrolling down. Or you can hold the shift and then press the page down option, page down. Then also it becomes relatively fast to select the lines. Now, as we know, we will delete till the atom section. We will not touch the heteroatom section. Okay, so you can do like this. So we have selected the entire atom of the B chain. Okay, then right click, cut. Okay, now it is removed. Now what we have is only A chain, and in the heteroatom section, right, in the heteroatom section we still have B chain present. That we will work out now. Okay, so now we have A chain. Now what that is what we did yesterday. When we have two chains, A chain and B chain, if both the chains are having homodimers, and if the ligand is not interacting with the B chain, so we will take only one chain, right? So that is why I am taking only A chain. I am deleting the B chain. That is completely based on this, because the Maravirok is interacting only with the A chain. Okay, that is why I am removing the B chain. Now, Maravirok as a structure we want. I don't need to draw the structure of this Maravirok because the coordinates, XYZ coordinates of the Maravirok is already given in the PDB itself. So I will just select this. Again, look carefully. Okay, MRV, wherever is mentioned, that is the Maravirok. A refers to A chain and the atom position 1101. And it goes till this. Then we have like Zn, that is the zinc ion. So I should not touch the zinc ion. That is not the interest. Then OLC is there, all those things. So that is not our interest. So cut the Maravirok, open the new window and paste. Okay, you don't need to cut that one. Paste a new file where you will have the Maravirok being present. Now why I did this, why I created a new file is because we want the Maravirok.pdb. It's a ligand file. Then only I can allow this ligand to interact with the protein. Now to bring the analogy, whatever we are doing yesterday, I have a lock, and to this lock already some key is bound to it, right? So to check whether the key is going to work or not, I have to, and to get to know what is the energy required for this key and lock to have the interaction. So I got to remove the key from the lock. Then you again check the energy that is required to open or to close, either of the things. How much energy is required? To test that one, I got to remove. I had to remove that one. That's what we are doing now. Okay, so I am removing the key and the lock complex was there earlier. I removed the key. Key is the Maravirok and separating it as a new file. And you have to save this file, save, and save this one within the same folder. Okay, as MRV, okay, .pdb. Save this one as Maravirok or MRV .pdb. Then save it. Okay, now we have MRV. Are you done with this, or is there any doubts associated till this step? Anyone? No. Okay. Now we will go back to this 4mbs.pdb. We want only the protein. We do not want any other aspects of the protein. And we, after looking into this leak plot, Meravirak, nowhere the water molecule interaction is there. So water is not required. We are not able to see the zinc, so the zinc is not required. The Meravirak is not interacting with the OLC nowhere, so that is also not required. So what we want is only the protein. Let's select everything. Okay, select everything till the end. Water is not required. Connect, master, nothing is required. So select everything and then say delete. Okay, select everything and then delete. Now what we have is only A chain being present. No heteroatoms are there in this file. Okay, only atom is in for this is all comment section. Okay, so you don't need to worry about this. Even if we delete this, still we will be able to see the protein. This is just the description or called like a remark. What is important for us is the atom. So we have all the atoms, corresponding proteins, XYZ coordinates and everything. Now this file you should save as. Okay, don't make the save, because don't save it like 4 MBs. We are not having the same file as the PDB, whatever we downloaded. Now you make it like _A. Okay, better, it is always recommended to have the file name as a single. Okay, don't give anywhere the space. So don't make it like 4 MBs space A. So make it like _A, so that everything needs to be connected. Okay, then we won't get any errors when we execute. So the moment I see like 4 MBs _A.pdb, I will get to know like it has only A chain. It is not the original file, so the file got processed. What is the processing done? Meravirak is removed and placed, kept as a one file. All the heteroatoms, water molecules, ions, whatever is there, removed. Right? So keep this file. Now, if we go back to our folder, we will have 3 files. Now, 4mbs.pdb is the original file. 4mbs_a.pdb is the only A chain. Meravirak.pdb is another file. The ligand, this acts like a protein. This acts like a ligand. Now, if we want more ligands, right? If we want more ligands to test, just like whatever we worked out, L1, L2, L3, L4, right? We can always take those ligands. Or if somebody has given you, right, synthesized molecule and then given, you can use that. Otherwise, you can, this is completely left to a researcher. Whatever, if a collaborator has given, you can take the ligands, whatever the collaborator has given. Or if somebody says like it is better if you use these ligands are having anti viral, use that. So it's left to the researcher to use the compounds. Okay, so there is no strict rule. You can download the compounds from the PubChem also, zinc also, or Campbell also, whatever you feel like one can use. Let us say the Meravirak is already we have used, right? So the Aplavirak is there, another molecule, right? So let's see how and where this ligand is interacting with the protein, where it is interacting. If that is the case, right, we can download this molecule. These are the related ones. Similar compounds. Cross references to other databases, PubChem, zinc, right? Lipinski rules. That is one violation, okay, because the molecular weight is 577, so which is higher than 500. So that is one of the violations. Okay. Now, if the 3D structure, if not available directly in this, anyway, we have a hyperlinks to other databases, right? So we can.
Sir, I have a doubt.
Yes, sir.
Sorry to interrupt you.
No
So
problem.
I tried using PubChem. This was one of the neurotoxin that I tried to download, and it did not have 3D structure. It had only the 2D form. So can I use that and work with the PYRX?
Yes. So let us say if you have only 2D like this, and if
Yeah.
the 3D is not there, we can convert the 2D to 3D and then use it.
Okay.
Okay.
Okay. From the
So
secondary
now,
structure to the tertiary structure,
yes.
and then.
So let's see if I will do both the things. So if I select the 2D and say download and SDF, okay, download this one. This is 2D getting downloaded. Save this. And let's select the 3D. This is the 3D structure, all right, where we can rotate. Then download. And this is the 3D for the same. Save this. Okay. Now, like this, if you want to take another ligand, okay, so that is how you will keep building the number of ligands. You can keep adding. So for our P51681, okay, we took this one now, apply rock, right? So this is also, let's say how is this ligand is going to work. So you can take that. Again, this is in the phase 2, that is fine, and as an antagonist, that is fine, and hyperlinks to other databases. If it is there, yes, it is linked with the Campbell, sorry, PubChem. So you can right click, open. Let's see if I again download the 2D of this file, save as TF, and 510 is the accession. This is the 2D. Download this file, then select this 3D, download SDF, download this file. Okay, so like this you can keep adding the ligands. Okay, so I think 3 ligands we will work it out. Now when I go back to our folder, right, so on the docking, now I have original 4MBS A with only A chain, then the Maraviroc is there in the .pdb. Then we took 2 ligands, one ligand with 2D and 3D, another ligand also 2D and 3D. Okay, so these are things we have now. Then once we have this, now you open the PYRX. Okay, so just type in the Windows. You might have the shortcut also in the desktop. Click on that PYRX. It is going to take some time. Goes with the RAM speed. Okay, so 64-bit or 32 version Ubuntu or Windows. So usually it takes around 1 minute to open. Slightly heavy code. Is it open for anyone? The tool?
Yes, sir, it's open.
Okay. It's still open. Okay. Now, once it opens, this is how it looks. Okay. So we will have right. So you will have it something like this. Maybe we will not. Your molecules, nothing will be there, and then we have like Autodoc, then one panel showing for the ligands and another panel showing like macromolecules. Okay. Now these are the two. Let me delete any previous. So all these are different projects. Okay. So different projects that. So you don't need to worry. So if you look into right, everything most of the time it will be saved with the PDB ID only. And here again there are few ligands that we are working with. You don't let it be like that. In your case, since it is a first time, so everything will be blank. Okay, so you won't have any ligands, you will not have any macromolecules present. So that is fine. Now, once you have this, sorry, just here. If you in the bottom, right, we will have it like Vina wizard, Autodoc wizard, Open Babel, Python shell, or Logger. Okay, there are two wizards that are there. One is called Vina wizard and another one is called Autodoc wizard. So there is a small difference between these two wizards. Vina is a program and Autodoc is also another program. Vina is fast, okay, and fast and accurate, and but provides less information in the sense we will not have it like ligand efficiency, other aspects. Now, even if you recall the presentation from Dr. Karthik the last Saturday, right? He also demonstrated the Autodoc. So he used MGL tools as one of the program. Then where we have like macromolecule open. Then we went for the command line. Then it was typed like Autodoc autogrid 4 -p, the file name, GPF, and then into GLG. Then Autodoc 4, the file name, something .dpf, then into DLG. Right, so it was a command line based what was demonstrated. So this PYRX is a graphical user interface where we don't need to type anything. Okay, so the process is automated. So it is going to do the things automatically. So you don't need to remember that -p, -l, or calling those autogrid, Autodoc, all those things is automated now. Okay, that is why it becomes very easy to perform docking with this PYRX. Now what you have is a 0.8 version, but I have purchased the version 0.9 version. Okay, because there are some advantages, and since I do the docking on a regular basis, and there is also some modifications that is going to happen in the process of docking when we are doing. So those things are supported in the commercial version 0.9. So purchase this, and I am using this 0.9. In your case, it will be like 0.8. The way it is going to execute the program algorithm that is going to work remains the same whether we use 0.8 or 0.9. Okay, there is no difference in that. Only there I can just tell you some of the advantages with the time when we are executing. Okay. Now otherwise everything looks exactly the same. You will still have like open babel, all other features remain the same. There is no difference. Now you click on this open babel, okay, then say this plus symbol, insert new item. Now this open babel, okay, docking. Open babel is for the file conversion or for the energy minimization. Even if you can remember the presentation, whatever Dr. Karthik has made, it was clearly mentioned that the energy minimization is a key step that has to be done so that we will have a right conformation for the ligand or the protein. Okay, so energy minimization is a key step so that you will, if there are any bond length, bond angle, torsional angle, if there is any violation or if there is any strain, if they are not correct, right? When you do the energy minimization, so all those issues will be taken care by the energy minimization. So if you just want to know how exactly the energy minimization is going to happen, okay, so I'm just going to take one particular example. Maybe we can also write the structure here. Okay, I'm just doing something in random. Okay, let us assume like this the structure. Okay, now the bond lengths, bond angles are appropriate. Let us assume like when we have a macromolecule or whatever, when we do some processing, let's assume like we made a mistake by placing the atoms in a wrong orientation. This we placed it here, this we placed it here, this we placed it here, right? Let's assume like there are some mistakes happened. Okay, I made too many mistakes just to just for you to see the changes that is going to happen in the structure. Now here also, let us say the bond length, whatever is supposed to be there, right, 1.76, let us assume. What if when we are making some changes, we reduced from 1.7 to 1.2, which is not correct. Okay, like this, we might have done too many mistakes in the, too many mistakes when we are preparing the molecule, right? Now, overall, if we look, right, we feel like the ligand or the structure is correct. So the number of atoms, right, and also the way it looks, everything looks fine. That's what our initial feeling will be. But the bond length is not correct, bond angle is not correct in many of the cases. So the process of energy minimization refers to whatever the ligand, whatever the atoms we have, all the atoms should maintain the appropriate bond length, bond angle, and torsional angles. Now, what is that minimization refers to? Let's say molecular mechanics, minimize energy. If I click, okay, I will place it somewhere around here. The moment I say run, whatever the mistakes happened in the ligand, bond length, bond angles, whatever, everything get fixed. Okay, now if you observe critically, I will just say run. It is going to happen quickly. Okay, within few seconds of time, everything gets fixed, the problem, right? Done. Now, bond length, bond angle, this is back to 1.53, 1.53. What was there earlier was 1.73. right? The bond length, bond angle, everything is fixed, right? This is how it was initially, the incorrect structure. Then when I performed the molecular the energy minimization, right? I'm just showing you the step that happened within a seconds of time. But if we want to see the changes that happened, I am playing it now here. First, the relaxation of the bonds happened. Then the relaxation of the side chain. Then this is how each step, the process, as entire process it has run, right? So probably within 8 or 9 iterations, okay, so that also we will get from the details, how many iterations. So iterations, there are 56 iterations that has run, okay, from this step to this step, there are 56 iterations, and it has worked out with the stretch, bend, stretch, torsional angle, non-bonded, everything, and then minimize the structure. So if we use a more complicated structure, okay, so the relaxation is going to take more time. But yes, the, let's say if I take any ligand, for example, I know I'm opening it in wrong window. Did I close that? Okay, I'll just open Chem3D. If your molecule is more complex, okay, then the time duration that it is going to it's hanging actually. Okay, so you got the concept, right? That's more than enough. So each step, it is going to take some iterations. Don't worry about the number of iterations, but the final aim is to get the optimized ligand. Okay, that's the process of so that energy minimization can be done using online servers also, like ProTruck, or if you have a commercial tool like ChemOffice, there also you can do, or using PYRX also we can do the energy minimization. So I will just show you, because if I use the energy minimization in the PYRX, right, the moment whatever we observed, the atom movements, that is not easy to identify in the PYRX, because here it happens in a much faster way. Okay, so let's go to this open Babel, then select this insert, then go back to the folder where the docking is there, and then let's say we will take the 2D. Okay, now if I select this 2D and open, we have the ligand now. This is the 2D structure. If you want to see the difference, so the between the 2D and 3D, okay, now this is 30013. If I open the 3003D, okay, so this is the 3D structure and this is the 2D structure. Okay, that means the energy is not optimized again, or energy is not minimized. So everything is there in the one plane, but if it undergoes the minimization, the folding is going to happen, the bond length, the bond angles, whatever is required to be maintained, that it is going to be maintained. Okay. Now, if this is the 2D. Let's take another 2D structure, whatever is there. This 300 is one, then 510 is another ligand. Now open this also, 51039. Okay, here you are going to get only the PubChem ID. This is also a 2D. If I open the 3D for this one, 510, right? So this is the 3D structure. This is how it has to be, but what we are having is something like this. So as one of the question, like what if we do not have the 3D and if we have only the 2D, something like this, what should we do? So open that ligand in the open Babel, okay? Then whatever the ligands you want to minimize, select those ligands using control, then right click. So this I want first, okay? So right click, say minimize selected. Otherwise, we can also use the minimize all, okay? All the ligands, whether it is a 3D, right, or a 2D, whatever the molecule we have, it is going to undergo the minimization. So since it is from the PubChem, it is better to minimize all the ligands. The energy that requires for the energy minimization to happen for the 3D will be less as compared to 2D. Okay, 2D will take more energy for the minimization to happen because it's not in the right order at all. More iterations has to happen. Then right click, say minimize all. Okay, so this is also going to happen in a very quick amount of time. All 4 ligands got minimized in a very quick amount of time. Now, as I mentioned, the energy for the 2D, this was in the 2D, this is the 3D. So 3D structure, energy minimization is faster and also takes less energy. But the 2D to 3D when we do, right, takes more energy. Okay, this requires 7,788 calories for the minimization, but the 3D only is 598. This is again 23,000 some universal force field, UFF. That's the force field used for the energy minimization to happen. Then again, this is only 640. Since okay, we can use any of these things. Okay, so there is no issues associated like which one to use. But when if there is a possibility of getting a 3D, okay, always use that 3D. Okay, in the PubChem or wherever. If there is no way you are going to get the 3D, okay, then you get the 2D and then do the minimization. Now this also we can apply for the further minimization to happen. Usually the first time minimization is enough, but is there any further refinement is going to happen for this? If you want to take, right click, minimize selected. Okay, then it is asking, say okay. Now if you observe, right, so the further optimization of the ligand is happening for this. I hope you could observe the rotation that happened. Now what made me to think of doing the further refinement, right? What is the reason? Now I will show you this one. Now if we look into this, right, this is not the right conformation for the ligands. It looks like conjugate. Here also the terminal atoms, if we look into, right, the positions or the orientation of these atoms are not proper. How can we get to know, right? If we just take the same ligand with a 3D, and if we look into, right, now this is like the angles are well maintained. Angles are well maintained for all these things. How about if we take the same ligand but convert it? Right now, if you look into, right, how is this and how is this? This is not the right conformation. Again, this is not the right conformation. This is not the right conformation. Right. So what I should do to get the right conformation? Take this one, ask it to undergo the minimization once again. Okay. We will have it something like this. Keep everything as it is, conjugate gradient, whatever is there, keep the default. Now, if you observe these atoms, will go a minimization. Minimization means the length, bond angle, torsional angle, everything gets relaxed. Now everything is having strains. This is not the orientation that it has to be. Say okay. Now if you observe, right, see how it is getting relaxed by itself. So this got relaxed. This region got relaxed. But how about this? It is still not completely relaxed. Again take it, minimize, then again say okay. Further refinement. So how many iterations to happen? That's a major question usually in the energy minimization process. Okay, how many such iterations? Initially, if you observe, it was 23,000, second iteration 17,000, 786, 17,340. If I take this one, initially it was like 7,000, then it has come down to, now it has come to 2,555. Is there a further refinement that can happen? Again, take it, give it, again further refinement is happening. Now come to 2,281. Now, where to stop, right? So that is why we usually won't go with this 2D. Okay. Again, if we look, still it is not reached to the conformation, whatever we want, in the sense, because from our chemistry background, right, you can easily make out the structure is something like this, right? If I have to place it, both the things, this is the fused ring system, right? If I keep the fused ring and then look at the energies for this fused ring, right? The atoms, the bond angles, completely relaxed, everything. But from the 2D, it has not come to that state. So if it requires, again minimize. So to avoid all these confusions, if by chance if we are getting a crystallographic structure, right, we prefer to use that crystallographic ligand structure and do the minimization, or preferred to take the whatever is available from the PubChem. If we are getting the 3D, right, better to download the 3D and work. If it is a 2D, there is some challenge with the SO challenge with reference to. What we can do is we can delete the ligand which is from the 2D portion. Okay, that is here. Delete current item. This into mark. I do not want this one. I do not want this one. With reference to nets, and overall it looks fine. Though you may get another question, what about the Maravirok? Right? Whether Maravirok should also undergo the energy minimization? No, we usually won't take the co-crystal structure because the Maravirok coordinates, whatever we have, is from the So that is why we will not take this ligand Maraviroc for the energy minimization. Okay. Now whatever we have it, right? Here, is it audible?
Yes, sir.
Okay. Screen also visible, right?
Yes, sir. Now screen is visible.
Okay. So, okay. Now that was a tedious process. Now because of this GUI, graphical user interface. Okay. Now we have two ligands. We do not have added the macromolecule yet. Okay. Now we will use this Vina wizard. Okay. Let's select this Vina wizard. We have two options here. One is called local and another one is called remote. Okay. Now whether you want to run within that laptop, whatever the RAM is there, the core, within this it is going to happen. If I select remote, so the files will be sent to some other person, okay, or Amazon Web Services or high performance clustering. So we can give those, we can submit, we can send the files to their server, and then the docking is going to happen there. Both options they have given. Since we have only 3 ligands, so this option of remote will be used when you have like 1 million, 2 million compounds. When we have like 500, 1000 ligands, the remote is not required. So local should be fine. And also, if your project is, let's say, what kind of a work you are doing. So if that is the case, people go with the local only. So you don't want to share any of your files to anyone, right? Remote is something like you are going to share the files to their server. So if you don't want, most of the time people prefer to run it locally in their own system. Okay, you don't want to give it. Then this is the first step within this wizard. Okay, local. Then click start. Here two ligands are selected. Okay, don't worry about that one. No macromolecule selected. In your case also you will be having only these two ligands. There is no other ligand found. Then we have an option here, add ligand or ligands. Click on this add ligands. Then go back to your folder where we have this mrv.pdb. Then you click this mrv.pdb because we have to add the ligand, right? Now the mrv gets added into your ligand section, right? Now we will have 3 ligands will be present. Now say add macromolecules. Now we should select 4MBS or 4MBSA.
Yeah.
Yes. So we had to select this 4MBSA because we want to take only one chain where we do not have any heteroatoms present, no inhibitor like Maraviroc, OLC or zinc ion present. So we have to select this A, okay, 4MBS A, then open. Now it is going to load. Automatically the okay. So if you want to, you can take 2 minutes time, okay, and you can download and upload the files. So you can just take 2 minutes. If I'm going a bit fast, you can just quickly go through this and then you can upload the files. Yeah. If there are any doubts or anything, you can let me know. So how many of you could upload the 4MBS A and also the Maraviroc? If not, with all the ligands, if you have Maraviroc, that is also enough. Don't worry about the other two. Okay, if you can work with one ligand, whether 2, 5, 500, 5000 or 5 lakh, not that difficult. Okay, so one protein, one ligand docking, we should have this C33 or C25, these two ligands. If we have 4 MBS and MRV, okay, that's more than a
Sir,
yes, sir.
sir, for me in the ligands it is showing only MRB, and in macromolecules it is showing only A chain.
That's more than enough. That's what only MRV, and if you can see only this, that's fine. Don't worry about you don't. We will just select additionally. Okay, so we will keep selecting like this if we want more. But it is holding the control button. But when we have one, this is enough. 2 is more than enough. One ligand and one protein is fine. Good. Anyone else who is doing parallelly? Okay. Shall we continue? If you are there in this stage. Okay. So once we have this, okay, let me place it here. Now the next step is to select the ligands. If I want, we will just hold the control button and then press, okay, to select the ligands. If I want to select all the ligands, okay, we can select everything. Sorry, from here you can use the shift and select. Right. So 11 ligand, 14 ligands selected. Since our aim is to take only these three, okay, 2 ligands are there in the phase 2 and 3, one is there in the phase 4, and this is our protein. So this is fine. Now we will get that information. 3 ligands selected, and then this is the PDB-QT selected. Okay, so this tells us clearly what are the ligands selected for the docking, what is the protein selected for the docking. Then save forward. Okay, now this box, whatever we can see here, is referred as the grid box. Now, the reason for not using 2 chains or making it into a single chain is because of this grid box. Okay, so if I click this full screen, is if I have another chain present here, so our grid box becomes too big, something like this. Okay, then the interaction of the ligand becomes slightly difficult. The assumption here is, since A chain and B chain is identical, the way the ligand interacts with the A chain will remain the same for the B chain, because A chain and B chain are identical. That is why we take only A chain. Now, there are 2 types of docking that will be done. One is called blind docking, okay, and another one is specific docking. Now, blind docking refers to, this is the grid box. Okay, now what is the role of this grid box? Role of the grid box is, this tells to the ligand, okay, the grid box dimensions. I will make it again. We have X, Y, Z, that is this center. Then the dimensions X. Now if you observe, the Z value is changing. If I change this, Y value is getting changed. And if I change, I think this is the one in the background. Now the X dimension is going to change. Okay. Now this acts like a grid box, acts like a pin code. Okay. What is the use of pin code in our letters? Is we are telling specifically where that letter or a post should go to, right? So based on that pin code, it will reach to the specific city. Okay, we bring to the specific city where it should go. And we also know that this grid pin code is a unique for each city. In the same way, grid box tells that you have 3 ligands and all these 3 ligands should go inside this box. Now where they are, okay, one ligand is over here, just double click on that ligand, okay, then we will get to see. Now where they are present, the ligands are present over here, okay, the Maraviroc may be there inside, okay, but again you don't need to worry that the Maraviroc is interacting with the protein, okay, it is just there inside because the coordinates are same. Since the ligand is removed from the crystallographic, so when we open, it will show as if it is there in that. But since these 2 ligands, whatever we are working, right, they were not the complex, it was just downloaded and made it to visualize. So you can see like they are present somewhere in this particular screen. Now, what is the way by which we can make these ligands to go inside this box, or what is the way by which we can make this ligand interact with this protein, is through this grid box. It is something like you are posting the letter somewhere in some corner, but you want that letter to reach to a specific person or should go to a specific post office. How is it possible? give a pin code. Okay, then wherever it is there, finally it will reach here. So let's assume like this is your city. Okay, and this is where this person should go. Go to Chennai, right? Or the place name you are telling. That's what the grid box is going to do. Any doubts with this? Any doubts with this grid box? Now, what if I do not want, or in other words, I know where the Meravirok is interacting, right? I know where the Meravirok is interacting. I know the binding site region already. So why should I give a blind docking to happen? Is it not possible that the ligand should interact at a specific site? Can't I make it something like this? Because I know where the ligand should interact. The binding region is very clear for me. I have collected that information from the PDB sum. I know the residues. So why should I do a blind docking? Then set the grid box. If you know the binding site, don't do a blind docking, set the grid box. Okay. Now in this view, right, it becomes too difficult to know where are those important, that is, those residues are this one. These are the residues where the Meravirok is interacting. That's why I asked you yesterday to make a note. Label these residues and we can see where they are present in this structure. Now maybe one person can just tell me the positions, okay, amino acids. Okay, now whatever you are seeing here, right, this is the protein. Whatever the line representation is there, this is the line representation. Everything is amino acid. This is one amino acid, second, third, like this. So this is how it represents. In the purchased version, we have a option. I don't think it is the same in the free version. We can make it like ribbon. We will. So we can make it like a ball and stick. There are different options that are there. Okay. Now let's remove this ribbon. Go to this molecule section. Initially the molecules, nothing was there. Now whatever the molecules that we are, those things, it is going to show it here. Now if I click this plus symbol, it is going to show all the amino acids that belongs to this particular protein. All these are the amino acids. And now we want to see our binding site residues. Now we know that the ligand is forming two hydrogen bonds. Can you tell me those hydrogen amino acids where the hydrogen bond formation happened? Anyone? Meravirok formed hydrogen bond with two amino.
So it is tyrosine nitrogen with oxygen 251 question, sir.
If you want, you can go to PDB sum, type for MBS, search it out, and you can tell me. Is there anyone who made a note of those two residues where the interaction is there? So which I mentioned yesterday. Yes, sir. Anyone?
Yes, sir. That is tyrosine residue, sir.
Tyrosine.
Nitrogen, oxygen.
Yes,
A
perfect.
251 position.
Good. And then
The other one is in the 370 positions of nitrogen with oxygen. The same tyrosine density.
yeah, tyrosine 37
Yes.
and then tyrosine 251. Thank you very much. Perfect, madam. Good. Now, what you can do is select this tyrosine 37, okay, and then make it like display, label, atoms. In the 0.8 version, this ball and stick option is not there. I think you will get only something like display, labels, select that. And then the one more hydrogen bond happened with tyrosine 250. Scroll down and then check whether in the 251 position, whether the tyrosine is there or not. If by chance in the 251 position, if the tyrosine is not there, TYR, if it is not there, let's say if alanine or some other amino acid is there, then the loaded protein is wrong. Okay, that is the way you can cross check whether you are working with the right protein or something with the wrong protein. We made it like right click, display, label, atoms. When we make that one, right, we can see the labeling done for those two amino acids. Here are those two amino acids where the labeling is done. We got to zoom in. Then you can see this is one, this is another atom, another amino acid. You can also select the hydrophobic amino acids, few hydrophobic residues. Anyone where those hydrophobic interactions were there, at least 5 or 6 residues we will select so that our grid box becomes perfect. Anybody? This is a key aspect in the entire molecular docking. Okay, grid box setting is the most. It's something like when you write a letter, writing the pin code is the correct method. What if you write a wrong pin code number, then the entire letter, whatever you have drafted, is a waste. Okay, so that is why I'm giving, in most of the cases, I give the analogy so that you can easily understand and appreciate why that particular step is introduced in the pipeline or in the process. If the pin code is something like 560085 for the PES college, what if you miss 56008, if you type 5, if you think like overlooked or felt like not required, right, it is not going to reach. So in the same way, everything is important. What if I simply put the grid box here and do the docking? Then the ligand will go here and start interacting where the protein is not there. So you will not get the energy. Then the purpose of doing itself becomes wrong. So if we set a grid box, something like this, and then do the docking, then the reason for looking this binding site, interaction where it is happening, the two hydrogen bonds, this was not required at all, right? So when we have performed the thing systematically, right, let's use all the information that we have. Okay, anyone? Other hydrophobic residues? I have it here, but I just want to know who are all working or listening or making a note. Okay, just to check that one I'm asking. Otherwise, I have that window right away. Okay, but I just checking. It's good that there is one faculty or one participant mentioned the hydrogen bonds where it is. So that gives impression that yes, maybe making a note or searched or giving the response at an appropriate time. So others can also make an attempt, okay, and just quickly tell me the hydrophobic residues. Other than two hydrogen bonds, all other amino acids are hydrophobic. Others?
283 position.
280. Glutamic acid 283. Fantastic. Display, label, atom. Okay, so that is also get highlighted somewhere. Okay, to see those things, I can reduce the grid box now. Since in my new version, right, I can make it like so. That's the advantage with the commercial. Okay, otherwise also we can work, but it is just showing with that ball and stick now. That becomes easy to other amino acids. I will deselect. Okay, so that my page remains same whatever you are working. So that is why other amino acids quickly. Others 284, and then any residues we can take 194. But the challenge is only one amino acid happens at a time. Okay, if I select 2-3 amino acids using control, we may not be able to make it like selection. So we have like 194 is there, 195 is there, then 198 is there, isoleucine 198, then 255. Okay, so when you are doing a project in your logbook, you can have a notes of all these things. 279. So that's the way which you can learn this. Having a book and pen and writing all the steps. Even I have learned with the same procedure. Since I did many times, so without looking into the manual, I'm doing. Otherwise, maybe for first 50 or 60 times, it is by looking into the entire protocol only. Phenyl label 112, then 109, phenylalanine. Right, when you are doing the real-time project, okay, it is better if you select all the amino acids that are there, whatever you obtained from your PDB sum. So 248 is the next one, tryptophan 248, label, atom, and then 284, 284, right? So what I'm doing is I'm just looking into this, okay, then selecting the residue. So 284 is done, then tyrosine 89, okay. Now we have selected the atoms of the amino acids. I think we have covered more or less all the residues. Leucine 255, yeah, it's done. Now this is the region. If you want, you can hide the line representation also. Okay, now we will have only the amino acid labeling made. That means these are the residues, amino acids, where this Meravirok is interacting with. Now we should put our grid box within this place. Okay, now set the grid box. Previously, whatever Dr. Karthik has demonstrated, it was not something like you can move the grid box as we are doing now. There was an option, okay, where you got to keep dragging manually. So when you drag continuously the x-axis, like scrolling, then the x-axis was moving like this, the grid box. Then when you drag the y-axis, then the y-axis will move like this. When you drag the z-axis, right, then the z-axis will move like this. So then there is one more option for this. This you got to change, then this would change. Then if you change this y, then the y would change. If you change this, then this would change. Again, drag this to center. So the setting up of the grid box itself was taking around 1 hour of time. Okay, literally 1.5 hours activity is there just to set this grid box. So that time taking task is now reduced. So wherein we have an option to move the grid box, right? Just use your mouse and then we can set the grid box within few minutes. So that's the advantage that is there with this PYRX. Otherwise it's a manual process. Now make sure in the 360 degrees, all the atoms are covered within this grid box. Okay. By chance, if you miss, why I'm mentioning is, by chance, let us say if you keep the grid box something like this, if you observe, these atoms are not covered, these atoms are not covered, these atoms are not covered. So what is the effect of this is the ligand will not interact with these atoms. In other words, this amino acid is not covered in your grid box. So the ligand will not interact at all. So to avoid such issues, set the grid box. Even if it is slightly increased more than the binding site region, that should not be a problem. But if it covers less residues, binding region, if it is not covering, then it is a problem. Again, it's as simple as if you give more details in your letter, right, that is not a problem. But if you give less details, then only we have a problem. Nearby, if you type something like the address near this location, landmark, right, or the bus stop, those additional information is not going to create a problem not to get that letter. But if you miss out the pin code, or if you miss out some of the important parameters, then the chances of getting that letter to your address is less. So that is why cover or be careful whenever you set the grid box. Now I think we have covered the grid box in all 360 degrees. Everything is there inside this. Now what we are telling is, let the ligands be wherever. The ligand should go inside this grid box and interact. Also, remember the Maravirok may look something like they're inside this grid box, but it is not the case. Okay, Maravirok is not there inside because the Maravirok is there as a ligand. Okay, it is not a part of the protein now. That means the ligand Maravirok will also be considered as a ligand similar to the ligands that are there outside. Okay, similar to this one. This will also interact with the protein. Any doubts with the step, whatever we have discussed so far? Anyone? Okay, now once we set up this grid box, okay, the next step is to click this forward. Now what are the ligands considered? We have considered these 3 ligands. Okay, so now the grid box setting is done, and then the moment I say forward, the ligands will start to interact with this. If we are doing with the Autodoc wizard, we have 2 separate steps here: run autogrid, then run Autodoc. Vina is fast because there is nothing like 2 separate steps to run. So when we set up the grid box, so the run autogrid will be done. Then the Autodoc is going to happen. Now when I click forward, when the difference, if I have to put again the autogrid and Autodoc, both the programs is going to work here in an integrated way. What I mentioned with autogrid is giving a pin code, right? It means where the letter should go. That is what the autogrid is going to do. Autodoc is once you are inside this grid box, what you are supposed to do. Okay, what should happen to the ligand when it is inside. It is something like me coming to the campus is a route, that is the autogrid. Now once I come inside the class, what I am supposed to teach or what I am supposed to do, that is what the autodoc is going to do. Okay, autodoc program is going to do. That is why we have two separate programs. Bring that guy to the campus is done by the autogrid, and once the speaker or the person is there inside this campus, right, what he is supposed to do, he is supposed to give a talk or has to demonstrate or to interact with all these proteins. In other words, this person has to interact with all these participants or the members, whatever. That is done by the autodoc. Okay, I hope you are clear with this difference. So when I say run, we know, or forward, now the process is going to start. So it is taking the first ligand and then the docking started. Then it will take the second ligand. Now if you look, right, analyzing the binding site, and random seed generation has happened. And also you got to remember that for each ligand, there will be 9 conformations generated. Okay, for each ligand, there will be 9 conformations generated. So maybe I will just quickly show you the entire process. In the meantime, when this simulation, I think it is going to take 2 to 3 minutes. By the time it gets over, we will look into this docking process. So the system becomes a bit slow because entire CPU is goes for the docking. So protein ligand docking, it's a molecular modeling technique which predict the position and orientation of a ligand. And the molecular docking problem is given 2 molecules with 3D conformation in atomic details. Do they bind to each other? Whether the protein and ligand will bind to each other. If they bind, what is its binding affinity, and how do the complex looks like? These are our challenges. We can do the protein ligand docking, protein docking, protein DNA docking, DNA ligand docking. All these are the different docking that possibilities are there. So the goal is, given a protein structure, we got to predict its ligand binding site. We can application, we can do for the ligand optimization or lead optimization, structure-based drug designing, virtual screening, high throughput screening, chemical mechanism studies. All those things are some of the applications. So when 2 molecules are there, ligand and a protein, how do the complex looks like? What is the energy that requires? That is what we are looking into now. That is the Venus score. Also the unit is kilocalories per mole, so considered to be a Gibbs free energy. So ligand receptor, when we put that one into a docking software, that is what we are doing. How is that complex looks like? Okay, and what is the associated score, that we will get to know. Same process, when we have a protein, multiple ligands, put all the ligands into this and then check where they are interacting. Then one of them may become like a future drug. This is the scope why we are doing. Now, how is the publication related to docking? So initially it was less. Then once the autodoc came into existence in 2004, 5, or even in the 2006 onwards. So there is a huge, because they made it open source. Okay, everybody can start using. So even in my master's project also, I used in 2008 itself the autodoc. So the number of publications are very huge now per year. We are having like 7,000, 8,000 highly cited. Which program, what all the different programs available? So we have like Autodoc, Gold, Glide, FlexX, Autodoc Vena, MOE, and HadDoc, other docking softwares. But widely used is again the Autodoc. You can find the papers even in Nature, Cell, Science, every journal, publishers, where the Autodoc is being cited. Okay, so that's why widely used program. This we have discussed even yesterday. We need the structure to do the docking. If the sequence is there, do the homology modeling, do the structure validation, or download the structure if it is available, removal of the water. That is what we did. Any heteroatoms, if it is there, if it is all correct, take that as a protein. Look for the small molecules, do the energy minimization. That's what we did. Universal force field, that is what we applied. Then we generated the 3D conformations, use it for the docking. That's the entire procedure. Now, how is the energy related? What are the possible poses and conformations available for the docking? When we have a protein, the ligand may sit here if it is a blind docking. What we have done now is we have asked the ligand to interact within this region because we know the grid box or we know the binding site. If we do not know the binding site, right, we can, if we set the grid box to this entire protein like this, then this is the protein. The ligand may interact over here, the ligand can interact over there, ligand can go and interact here. Now, how many such conformations it is going to generate? So Autodoc is going to generate 9 such conformations. For every ligand, for each ligand and protein, the ligand is going to interact with that protein 9 times. Okay, it's a prediction what we are doing. So whenever I put the structure like this, that's exactly this one. Okay, exactly like this. Now when I say the ligand, right, the ligand is going to interact with this protein 9 times. One time it will interact here, come out, change the conformation, go and interact with the other region. Third, fourth, fifth, sixth, seventh, eighth, ninth. So 9 conformations are there. When I set the grid box to the entire protein, it may go inside and interact once, go on the top and interact there, third, fourth, fifth. So we will have 9 energies associated for each ligand. So when I have 3 ligands and 1 protein, 9 into 3. So we will have around 27 conformations generated. Okay, 27 conformations. Now, sorry. Now the ligand is interacting here. We will have 1 associated energy, associated energy, associated energy. So this is referred as 3 degrees of freedom, relative position. If it is interacting, if we set within the grid box, so the 3 orientations, 3 degrees of freedom, it may interact like this, it may interact like this, or it may interact like this. Here only 3 representations are there, but in fact we will have 9 such orientations. Okay, 9 such orientations available for the ligand. One can give the flexibility for the ligand. So that is the n degrees of freedom. Then the ligand may take this conformation, ligand may take this conformation, because we have 3 degrees of freedom. That's the rotation that is allowed. Because flexibility, we usually keep the flexibility for the ligand. It can have a rotation. But for the protein, we usually avoid giving the flexibility because computationally it is challenging. Otherwise, we can keep the flexibility for the protein also. Okay, so this side chain is having a moment, this is having a moment. So we can give the flexibility for the amino acids also. As long as you are not sure about the residue which is having a flexibility, don't assign. Otherwise, we can assign. So what we are doing now is a rigid docking, because flexibility is not given for only for the ligand we have given. Then we will have the energy. So if the ligand is interacting here, this will be the associated energy. Energy is high. When it is interacting here, right, this portion, if you look, the energy is going somewhere around here. When the interaction is here, the energy is very less. Now on what basis they are telling? The interaction between this protein and ligand is there only in these 2 contexts. Complementarity is less. But here if we look, the complementarity is relatively high as compared to this, but still not that much as here. But here if we observe, the ligand is covering entirely. maximum complementarity is there between protein and ligand. So that becomes our least energy. E here is the energy, N here refers to conformational coordinates. Then we have the energy associated. So this is how, what even Dr. Karthik has shown, this is what is available, what was available. In few cases people do use this, but it is a manual process and slightly difficult to work with. Okay, so now that is why we started using PYRX. So we have a scoring function, we have a search methods available. These two are the most important aspect in any of the docking programs. How it gives the score and how it is going to generate different conformations. That is what two important aspects as far as the docking is concerned. Okay, now the docking is done. Okay, so I will continue with this the later point of time. Now the results, once the docking is over, let me close this. Okay, make it like this also. Okay, now if we want to see the molecule 4MBS, right click, display, maybe as lines only. Okay, now all the previous, this is the docking conformations, whatever we have, let's let it. Now the original Maraviroc is this one, and now 4MBS C33 is one ligand, the energy is minus 9.9. This is the second conformation for that. Okay, if you want me to zoom in. So for each ligand, C33 is one such ligand. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so these are the 9 conformations for one ligand. Then these are the 9 conformations for the second ligand, C25H40. And for the Maraviroc, again, these are the 9 conformations. 1, 2, 3, 4, 5, 6, 7, 8, 9. Now, if you look into, right, Maraviroc is having like minus 12.2 energy, while this C33 is having minus 9.9 and C25 is having minus 9. So minus 12 is much better than minus 9 or minus 8. So that is why Maraviroc is there as a inhibitor. Since minus 9 or minus 8 is also a good result, that is why they are testing it out. Okay, so now in most of the cases, our crystallographic ligands will have a better energy. Okay, will have a better energy like minus 9, minus 10. That is why even when I was explaining, right, minus 10, minus 9, like this only our energies will be. Now, this is what the entire docking is. Okay, so we can keep the results in one of the format called CSV. Select this, then save this file, vinanalysis.csv. Just save this particular file. Okay, so maybe tomorrow I will just show you how we can generate the Lig plot. Okay, and how we can decide whether the conformation of the Maraviroc with the minus 12 energy, whatever is here, whether the interaction of this complex is same as this or is it different. Okay, whether it is the same or whether the interaction is going for a difference, that we will analyze maybe tomorrow. It's the post-docking analysis that we will do it tomorrow. Now, any doubts with whatever we did today? For any participants, any doubts in the entire procedure that we followed? Once we get these results, the use of this PYRX is over. Okay, so we will use the other two tools. One is our PyMol. Okay, so along with that PyMol, tomorrow we will use one more tool called LickPlot. Where all these things comes. Okay, so you can also download this LickPlot. Just check if we can download. Like you got to academic license. If you do not have a password, please go to academic license and then you can get. And if by chance, if you are not able to get the LickPlot by any means, okay, I will provide my license file of the LickPlot and you can use it. But to use this, you need to have a Java. Okay, so install Java and then you can use it. Installation instruction, yeah. So if you go through this, it becomes very easy. So first you got to download this Java, install this, then install the LickPlot plus. That is just a, you will have it like lickplus.zip file. Okay, then if we double click, we will have the LickPlot open. So that is easy. Now one of the question is, can we use both the chains simultaneously for docking? We can use both the chains, okay, but we will have only one grid box. So rather than having it like this, we will have one more chain present, right? So two chains will be there. But for two chains, your grid box becomes a big one. That's all. Otherwise, but you will not get like two grid boxes, one grid box for A chain and one grid box for B chain. No, we will have only one grid box, which is going to cover both the chains. That is, what if the ligand is interacting at the juncture of both A and B chain, right? Then we have to take both the chains. So if that is the case, set the grid box in such a way that your grid box is going to cover few portion of the A chain, few portion of the B chain, and then allow the ligand to interact. That's the only way. Any other questions? From anyone? Okay, so if there are no doubts, then thank you very much. We will stop it here. Then we will, tomorrow, we will explain or look into post-docking analysis, how to analyze the results, how we can generate the LickPlot. So visit this, download and install the LickPlot. Okay, also install the PyMol, the newer version. Then it becomes very easy to execute.